Everyone, welcome back to the CRN Virtual Family Conference. So happy to have you here for day 3. We are looking forward to a full day with a series of talks from our industry partners and researchers. To make this easy on everyone, we'll stay on this link for this series of talks until we get to our panels later today. And so that is at 2 p.m. Eastern Time. At that time, you'll go back to the attendee hub and access those closing panels. So I just wanted to give a brief overview of what we're going to talk about today. We have talks from our industry partners, as I mentioned. We'll be hearing from Recordati, from Leadiant Biosciences, from Horizon Therapeutics, and from Avrobio. We also have talks from some of our CRN funded investigators, and that will take us into our panels at the end of the day, which will include a panel made up of individuals living with cystinosis and parents of individuals living with cystinosis. So we hope everybody is able to join us for all of those great topics. And with that, we'll jump right into our first presentation of the day. And that is a presentation from our industry partners at Recordati on Cystadrops, distribution, resources, and support services for caregivers and adult patients. This will be given by Helga Nemaric, who is the National Patient Advocacy Liaison for Recordati Patient Support Services, Candace Gafford, who's the Head of Insurance Services for Anovo Specialty Pharmacy, and Nelson Burford, who is a Doctor of Pharmacy for Anovo Specialty Pharmacy. So with that, we'll listen to Recordati's presentation.
Hi, my name is Helga, and I'm a Patient Advocacy Liaison, or PAL, with Recordati Patient Support Services at Recordati Rare Diseases. In my role, I provide a one-on-one personalized support for patients receiving treatment with Cystadrops, Cysteine ophthalmic solution, 0.37%, and their caregivers. I will tell you a little bit more about my role later on during this session. I am pleased to have the opportunity to host this session today. During the session, we will discuss how to get Cystadrops as well as the support services that are available to patients and caregivers. Recordati Rare Diseases, aka RRD, develops high-impact therapies for devastating rare diseases. At Recordati, we focus on the few, people just like you who are affected by rare diseases. You are our top priority and are at the core of everything we do. We are honored to serve the cystinosis community and provide medications and educational resources for patients and their families. I would like to start the session with information about how cystinosis may affect the eye and why your doctor may prescribe Cystadrops as a treatment. Cystinosis causes cystine crystals to build up in the cells throughout the body, including the eyes. The cornea is the front clear part of the eye and is the area where the crystals commonly build up. Without treatment, cystine crystals build up throughout the entire cornea over time. The cornea of the eye is avascular, which means it does not have blood vessels in it. Oral medicines that usually travel through the body in the bloodstream do not reach the cornea. So cysteine eye drops are needed to treat cystine crystals in the cornea. Cystadrops is a cysteine eye drop with a 4-time-day dosing that reduces cystine crystals in the cornea of adults and children with cystinosis. When Cystadrops coats the cornea, the active ingredient cysteine enters the cells in the cornea and breaks apart cystine. As a result, the buildup of cystine crystals throughout the cornea decreases with continuous treatment. It's important to note that Cystadrops should not be applied directly to your cornea. The Cystadrops drop should go in the space between your lower eyelid and your eye. You can create the space by gently pulling your lower eyelid with your finger. To spread the drop over your cornea, you should close your eye and gently massage your upper eyelid with your finger. Please see the instructions for use at www.cystadrops.com. The most common side effects of Cystadrops are eye pain or what might feel like stinging, blurry vision, eye irritation, but what might feel like burning, eye redness, discomfort at installation site, or what might feel like sticky eyes or sticky eyelids, eye itching, watery eyes, medicine deposits on the eyelashes or around the eyes. Cystadrops received FDA approval and has been available for patients since September 2020. Recordati partners with a specialty pharmacy Anovo to distribute Cystadrops. Anovo is the only specialty pharmacy that dispenses Cystadrops. The Anovo team members are joining us today. They will talk a little bit more about who they are and what they do.
My name is Candace Gafford. I'm the Director of Insurance Services at Anovo Specialty Pharmacy. I lead the day-to-day operations of payer services at Anovo. I work with insurance companies and physicians to help patients find coverage for their medication. I have Nelson Burford here as well.
My name is Nelson Burford, Doctor of Pharmacy at Anovo. I've been a pharmacist for 28 years, with the last 14 in the specialty pharmacy space.
Can you please tell us a little bit more about the Cystadrops prescription process? What can patients and their family members expect when they're being prescribed Cystadrops by their doctor?
Patients will receive a welcome call from Anovo, and then we will walk them through the process and what they should expect of the upcoming days.
Candace, many patients and families may get concerned about paying for their medication. What should patients do if their copay is too high or they can't afford it, or their insurance doesn't cover Cystadrops?
Sure. So Recordati Rare Diseases offers a copay assistance program for patients taking Cystadrops, as well as a patient assistance program for those without insurance. Anovo can help patients determine if they qualify and can assist with enrolling patients in one of the available programs. If you have any questions or a specific situation you'd like to discuss, please give us a call. Every situation is different, and we understand that, and we will be happy to work through your specific situation because our goal is to help you get Cystadrops.
Candace, please tell us what else can make the process easier and more efficient?
It's something simple as returning our phone calls. I cannot stress enough how important it is. Also letting us know if there are any insurance changes or any other circumstances that prevents you from continuing your medication. You can add our phone number to your address book or to your phone, so you'll recognize our calls. We understand that you may have a busy schedule, but please return our calls at your earliest convenience. This will help to ensure that you have Cystadrops when you need it.
What else? Do the patients and their caregivers need to do anything else to renew their prescription monthly?
No, the prescription is usually good for 12 months. When the prescription expires, the pharmacy team will contact the provider office to renew the prescription.
Wonderful. Let's talk about the pharmacy team at Anovo and the role they play. The pharmacy team at Anovo is responsible for shipping Cystadrops to the patients' homes and educating the patients on how to administer Cystadrops. Let's start with the shipping process.
Sure, Helga. We use overnight FedEx services to provide Cystadrops to our patients, and a signature is required to make sure it gets to the right person.
Nelson, can the patients choose the time for their delivery? And what options do they have if they're not at home at the time of delivery?
Yeah, the patients can choose a day and a place for the delivery that works best for them, home, a neighbor, or work. Packages are delivered within a certain time normally, and FedEx also has an app that they can use so the patient can track their shipment or even reroute their package to an alternate location like a local pharmacy for later pickup.
That's great. I'm sure many patients will find this app very helpful. Let's switch gears and talk about some helpful tips for patients starting on Cystadrops.
Everyone knows that administering medications as prescribed is important for treating conditions. When patients start on Cystadrops, our pharmacy team educates patients on how to assemble the dropper and how to administer Cystadrops.
I think it's also important to mention that for caregivers and parents of young children, it's important to educate other family members, such as grandparents, aunts and uncles. And let's not forget about the babysitter and your school nurse. Are there any other tips?
Getting into a habit of starting a new bottle of Cystadrops on the same day of the week will help you remember to discard an open bottle of Cystadrops every 7 days.
Thank you so much for such a useful dialogue. I hope that our listeners learn a new and helpful information and have a better understanding of how to get Cystadrops and what to expect throughout the process. I would like to recap all the services that you can receive through Anovo and then talk about the new program that's now available for patients on Cystadrops. First, the Anovo team will help you get started on Cystadrops. They will work with you, your doctor, and insurance company. They will help you determine if you need copay assistance and if you qualify for patient assistance programs. They will ship your medication to your home and renew your prescription every month. I did like to introduce a new program for patients taking Cystadrops. The PAL program provides one-on-one personalized support for patients and their caregivers that may include education about how cystinosis affects the eye, answering questions you might have about Cystadrops, connecting you to help or resources. El programa de apoyo al paciente y familia también está disponible si usted lo requiere en español. To enroll in the PAL program, go to www.cystadropspal.com, where you can fill out and submit an enrollment form. Remember, enrollment is voluntary. All of the services, including those offered by Anovo and those offered by the PAL program, are provided to you directly by Recordati Rare Diseases at no cost to you. The services are provided to help support you and your family while you are on Cystadrops therapy. For medical care or guidance, please continue to contact your doctor. If we haven't been able to answer your specific questions about Cystadrops and the educational support services that are available, please contact Anovo at 866-925-6212. Thank you so much for listening.
Cystadrops cysteamine ophthalmic solution 0.37% for topical ophthalmic use. Indications and usage. Cystadrops cysteamine ophthalmic solution 0.37% is a cysteine depleting agent indicated for the treatment of corneal cysteine crystal deposits in adults and children with cystinosis. Important safety information. To minimize the risk of contamination, do not touch the dropper tip to any surface. Keep bottle tightly closed when not in use. A condition where the pressure inside the skull increases for unknown reasons has been reported with cysteamine taken by mouth or cysteamine eye drops used at the same time as cysteamine taken by mouth. This condition went away with the addition of medicine that increases the production of urine. Contains the preservative benzalkonium chloride. Contact with soft contact lenses should be avoided. Remove contact lenses prior to application. Lenses may be reinserted 15 minutes following administration. The most common side effects are eye pain, stinging, blurred vision, eye irritation, burning, eye redness, discomfort at installation site, sticky eyes or sticky eyelids, eye itching, watery eyes, medicine deposit on the eyelashes or around the eyes. To report suspected side effects, contact Recordati Rare Diseases, Inc. at 1-888-575-8344 or FDA at 1-800-FDA-1088 or www.fda.gov/medwatch.
Please view full prescribing information and instructions for use at www.cystadrops.com.
Thank you for that from Recordati. Great information from Anovo as well. And if folks have questions for our partners at Recordati, you can send them to info@cystinosis.org, and we can always connect you to the correct individuals at both Recordati and Anovo. For our next speaker, we are happy to welcome Lesli King, who's the Director of Marketing for North America for Leadiant Biosciences, and she's going to give us an update on Cystaran. Welcome, Lesli.
There we go. Can you hear me now? Okay. Thank you, Christy. I'd like to take this opportunity to congratulate the CRN for continuing to host the family conference. It's challenging times, but you guys have done it and done it well so far, and I'm sure the rest will be fantastic as well. Thank you for inviting Leadiant to participate and provide an update to the attendees. And also thank you to the cystinosis community. I'm pleased to be here today to tell you all a little bit about Leadiant and to provide an update on the Cystaran shortage. A little bit about Leadiant. We work with rare dedication to develop therapies for patients with rare diseases. Our mission is to partner with communities, physicians, and researchers worldwide to overcome challenges and to be a resource of hope. We have a long history in rare disease, having received our first orphan drug designation in 1984, and it was the fourth orphan drug designation ever granted in the United States. It was for Carnitor, also known as levocarnitine, which may sound familiar to some of you. And since then, we've received 8 more designations, including one for Cystaran. For more than 2 decades, Leadiant has been working with the cystinosis community, and in 1988, we began collaborating with the National Institutes of Health and quickly involved the cystinosis community from the advocacy side. The collaboration with the NIH led to the development and commercialization of the first FDA-approved treatment for corneal cystine crystals, and in 2012, Cystaran was approved. So as you all may know, in October of 2020, we announced that Cystaran was going into shortage. And a shortage sounds like a product is just running low, but in the pharmaceutical industry and to the FDA, it means that we don't actually have the supply to fulfill the demand. Cystaran went into shortage because the independent manufacturer with whom Leadiant was working restructured its business under a Chapter 11 bankruptcy code, and that restructuring during the bankruptcy disrupted the continuous production of Cystaran. So what have we done? We've taken the following steps in order to restore the supply of Cystaran as quickly and as efficiently as possible. First, we identified a new manufacturer to produce Cystaran. We chose this manufacturer for their proven track record and expertise in ophthalmic manufacturing. We're confident that with our new partner, the product will be more consistent and reliable. And working to identify a new manufacturer is an important process, and we made sure that we chose the right partner for the future of Cystaran. And in order to address some of the past challenges with Cystaran, our new partner provides a proprietary technology that we will incorporate into the bottle's packaging, which we believe will reduce the likelihood of shortages and recalls in the future. We believe this technology will enable a more consistent and reliable manufacturing in line with the U.S. Food and Drug Administration's standards and requirements. Throughout this entire process, we've been working very closely with the FDA to ensure we're meeting their requirements for securing a new manufacturer, keeping them informed of our progress, and making ourselves available to answer any questions that they may have. So you might be wondering what's taken so long. Drug development is a highly regulated industry, and even the smallest of changes to a product require research, development, studies, review, and approval from regulatory agencies, including the FDA. And as you can imagine, partnering with a new manufacturer requires changes that need to be reviewed and approved by the FDA before we're able to begin production. So rest assured, Cystaran has always and will continue to undergo rigorous testing and quality assurance checks, both in the manufacturing process and in the final product itself. So what are the next steps? When supply is restored, there will be no changes to the formulation of the eye drops. You may be wondering when the eye drops will be available. The timing of the availability is not something we're able to provide at this time. There are still several factors in play, and some of those we can control and some of those we cannot. We can say that our activities with our new manufacturer, which have to be approved by the FDA, are in the advanced stages, and we're working really hard to restore that supply as soon as possible. Our commitment, again, is to the community, to the cystinosis community, which we've been committed to for more than 20 years. We know how much trust you put in us, and we're committed to restoring the supply and continuing to support the community. We're confident that we'll be able to deliver a consistent supply with the new manufacturer, and we will continue to provide updates regarding the status of Cystaran and when the supply is expected to be restored. In addition to Cystaran, Leadiant continues to explore new opportunities for improvements in the treatment of corneal complications associated with cystinosis. And we again just want to thank the CRN for hosting another robust and informative family conference, bringing the community together, as I mentioned, at the welcome session. It was incredible to see people from all over the world join. I think these times make it challenging, but at the same time, it's opened up a lot of opportunities for people to participate who may not have been able to. So congratulations to the CRN. We want to thank the community for your understanding and support through this process. and we're doing everything we can to bring Cystaran back to the patients as quickly as possible. We're committed to restoring the supply so that every patient in the U.S. who has a prescription for Cystaran is able to access it. And thank you to everyone who's participating in the conference. We look forward to providing additional updates regarding the supply as they become available. And as always, if you have any questions regarding your treatment plan, you should discuss this with your physician. And any questions about the shortage, don't hesitate to reach out to me. I'm Leslie King. My email address is leslie.king@leadiant.com. My phone number is 301-335-0019. Call me. That's my cell phone. So you can call me, you can reach out, and we'll do the best we can to help you. So on behalf of the entire team at Leadiant, thank you and enjoy the rest of the conference. Christy?
Thank you, Leslie. Thank you for that information on the issues with Cystaran. Currently, we're really grateful for your transparency, for the information that you shared with everyone, for sharing your cell phone number and your email. And Leslie has been a part of our community for as long as I have, so that's about 20 years. And we really, really appreciate her partnership and Leadiant's partnership in this regard. And we will keep the community up to date on what's going on with Cystaran and make sure that information is passed on. So we're actually running a little bit ahead of schedule, which is fantastic and something that doesn't ever happen in a live event. So I just want to remind everyone that when we're done with our industry updates this morning, we'll move to a presentation by Dr. Caterina Hohenfelner and her colleagues on the studies that they're doing that have been funded in part by CRN. We're going to go into other CRN funded updates from the lab at Dr. Fox and Dr. Mulholm and Dr. Francisco lead at Einstein. We'll have a break, then we will hear from Dr. Levchenko and Dr. Sarwal. And then finally, we'll end up the day with the two panels. First, the panel of parents of children and adults with cystinosis, and then finally a panel of individuals living with cystinosis. So we hope that you'll join us for the entire day. We have great information, and we want to be sure that everybody has access. As a final reminder, we'll stay in this link until the end of Dr. Levchenko's talk at 1:30, the end of Dr. Sarwal's talk at 2 p.m. Eastern, and then you'll need to log back into the attendee hub and click on the links for those two panels. So with that being said, I'd like to move to our next speaker. He is BJ Viau. He is the Director of Patient Advocacy for Rare Diseases at Horizon Therapeutics, and he's going to talk to us about resources for the cystinosis community. So welcome, BJ.
Thanks, Christy. Hey, everybody. As she mentioned, my name is BJ Viau. I'm the Director of Rare Disease Patient Advocacy at Horizon Therapeutics. Our team's relationship and commitment to the cystinosis community started back in 2016 and continues to grow each year. Horizon as a company, our mission is to deliver medicines for rare, autoimmune, and severe inflammatory diseases while providing compassionate support. This comes from our strong and simple philosophy to make a meaningful difference for patients and communities in need. And this video we're going to show here really just provides a bit of a background on our involvement in the cystinosis community and how we view our role as a partner. So after the video, I hope to highlight a few resources we made available for you. So cue the video.
Cystinosis is a rare disease, and in fact there are over 7,000 rare diseases. So there are treatments for only 5% of those 7,000 diseases. That is why we pursue what we do. That's why we continue to work hard to find medicines for these rare diseases.
We don't just come out with medicines to help treat the disease, but also create programs and services to help them through that entire patient journey.
Here at Horizon, working on the patient advocacy team, I am out in the community trying to learn, trying to ask questions, trying to better understand what the disease or condition might mean to you so I can bring back potential problems that some patients or caregivers might have and come up with solutions.
Something we heard loud and clear from the cystinosis community was the need for additional tools for ongoing management of the disease. One such tool that we developed purely based on the feedback that we heard from patients and physicians is the white blood cell cysteine level collection kits. This is available to patients at no cost, and we believe that it will help patients and their families better manage and better live with the disease.
One of the other things that my colleagues and I learned while being out in the community and listening and learning from all of you is the need for two things. Number one was the need for more education around cystinosis. And then the second one was the opportunity to connect with all of you. So that's where we created Cystinosis United, which is both a Facebook page, but also a website. And it's a place where we can post educational content, but also a place where you can connect with us. Another great benefit of Cystinosis United on Facebook is the opportunity to connect with other patient groups. You know, we post a lot of content on conferences and educational sessions going on in the community throughout the country.
Our approach is to better understand cystinosis. It's about talking with people. We go out to hospitals, we go out to practices, we talk with as many doctors as we can. Cystinosis is a complicated disease. There's a lot we know about it, but there's a lot we don't know. And in a lot of ways, our role in medical affairs is to learn what we don't know so that we can help inform our research, so that we can study the appropriate aspects of the disease, and so that we can learn more, work with doctors to communicate what we have learned and what we need to study. Pharmaceutical development and drug development is a very risky business. The risk is that most medicines that we try to develop or work on will fail. We are able to, in those rare instances, bring a medicine to patients that wasn't previously available. So this is a significant investment in people, in time, and in money, but the reward is that patients will have a medicine that they can use to treat a rare disease that at the individual patient level is so important.
In working with the cystinosis patient community, many patients and their families expressed how it is incredibly hard to navigate the treatment process. For us at Horizon, it is important that patients receive the medicine that their physicians prescribe for them. That is why Horizon has developed programs to help eligible patients get the medicine they need, regardless of their ability to pay.
In my short time in the cystinosis community, I've learned a lot. I've learned what it takes to be strong and to have the strength to deal with the disease on a regular basis while also trying to live the strongest, healthiest, most positive life. And the more I learn from all of you, the more inspired I am to go back on a day-to-day basis in the office and find new ways to partner and to find new ways to come together to hopefully one day eradicate cystinosis from all of our lives.
I was recently at a cystinosis patient event, and every day it's a reminder to me what exactly we do here, why we are here, and it's a reminder to me that when we're going through our day-to-day work lives, there's a cystinosis patient community at the other end that is hopeful that we can come up with services or programs that may help make their lives easier. We take a lot of courage and a lot of inspiration away from the actions of these patients and their caregivers.
All right, welcome back. So hopefully that video, helped you get a little bit more of a feel for who we are at Horizon, our goal of making medicines available while also trying to provide resources for all these other unmet needs that might be out there. And that's really what I wanted to talk about today. I want to share a little bit more of a few of the resources that we've made available beyond our medicine. All the resources I'm sharing with you are available to anyone, to everyone from the cystinosis community. And they come from that core belief you heard about in the video, to participate with, to listen to, and to learn from, those who are impacted by cystinosis, as all the pieces I'm going to talk to you about today have been created with the help and the guidance of individuals living with or in a family impacted by cystinosis. So really my hope for the end of this talk is, number one, you walk away with a new resource, either for yourself or someone else in your life. And then number two is, if you have any questions about these resources, about Horizon, that you have a friendly face that you can reach out to, and you feel comfortable reaching out to myself And if it's me answering the question or one of my team members, we look forward to helping you. So we believe in an open dialogue, as I mentioned. So anywhere throughout what I'm talking about today, if you have any questions, you can send an email to connect@horizontherapeutics.com. Just put in the subject line CRN conference, and that will get to me. So anything about these resources, anything about Horizon, I'll mention that email a couple times. So if you missed it there, I'll say it again. But as mentioned, 4 years ago, we created, with the help of the cystinosis community, the Cystinosis United brand, which is both a standalone website, cystinosisunited.com, and again, a Facebook page. And as the video said, the purpose of Cystinosis United is to provide straightforward disease education while also providing a platform to share relevant updates from community partners and us as Horizon. So we do our best. We try to bring creative, relevant content every day to the page, but we're always open to new ideas on how to improve. So if you haven't, give the page a like, a follow, or a comment on the chat if you already follow, and we look forward to connecting with you there. And as part of Cystinosis United, we make many hard copy electronic versions of this educational content we talk about, available at no cost to the community. Much of this content, it's vetted by the community, either families, patients, or physicians, and it's meant for you. So I'll share with you just a few pieces today that we've made available. And again, if any of these pieces seem to be of value, please reach out to that email, connect@horizontherapeutics.com. And we can mail you any of the pieces and any of the quantities you might seem necessary for yourself. So number one is Michael's Show and Tell activity book. This is a book. It's filled with fun things to do with kids while they read and learn what it's like for Michael to live with cystinosis. It's a great way to start a conversation with children to help them understand cystinosis, and it can be used in your home, or we've had a lot of people order a handful of copies, bring them to school to help classmates understand a little bit more about cystinosis. So you can order this book, or you can download it like I did, and you can print it off in black and white and color it. I still have some coloring to do, but it is there for you. Number two is the Self-Care Project Book. This is a resource for adults that can help you talk through how self-care relates to your cystinosis care and overall health. It's about setting goals. It's about carving out time for things you enjoy and prioritizing the number one thing, which is yourself. Piece number four is our BU booklet. This is a fun and interactive booklet that helps others with helpful reminders, with tips and tricks to live your best life. It's a resource for teens that has tools to support you as you juggle life, school, friends, and cystinosis. And then lastly, we have Ruby's Quest for Crystals. This is a book where Ruby, the main character, she goes on an adventure inside the body to learn about cystinosis and crystal buildup in many of the body's organs. It's meant for parents to use with their young kids or others looking to learn a little bit more about cystinosis and how it impacts the body. And we got a lot of great praise on the Quest for Crystals book. So I'm really excited to share that. We've actually announced that Ruby's Quest for Crystals storybook is being turned into a short film for the community. And to put some kind of fun around the video premiere, we're hosting a few virtual activities the week of August 2nd and invite all of you to participate. The week's going to consist of a craft night, a trivia night, a panel session with advocacy groups, including CRN. And Friday night, August 6th, is the movie premiere. I believe it's going to be about a 10-minute long video. As I mentioned, anyone is welcome to register. All participants, they're going to receive a box at no cost that will have a host of items to make the week fun and interactive. The content's meant for kids, teens, their parents, but really anyone in the community is welcome to participate. So if you, your family, your friends, you want to participate, you can reach out to that email, connect@horizontherapeutics.com. You can put in the subject line CRN conference or Quest for Crystals, and you'll get a response to make sure that you are properly signed up. So excited for that. Hope to see you all in a few weeks with Ruby's Quest for Crystals movie premiere. And then the last resource we talked about in the video was the importance of the WBC kits. This is a resource that we really learned was needed from the cystinosis community. was just an easier way to get these kits to you and get the results back to you. So as most of you know, white blood cell cysteine testing is recommended to figure out how well a cysteine depleting therapy is working. And Horizon helped bring these kits to life in 2019. So they're still relatively new. And we did this through the support and feedback of so many families and physicians. And a couple just simple facts about the kits is they must be ordered by a physician. The kits come at no cost to you, no cost to your doctor, no cost to your insurance company. Horizon does not receive any patient confidential data about these kits. The tests can be sent to either of the two labs, either UCSD or Baylor. And for more information about the kits, we do have a website. It's wbckit.com, wbckit.com. And it really has a lot more information for you about the kits in a very simple manner to help you get these and get the results that you need. So hopefully those can be a resource or continue to be a resource for many of you out there. And those are really the core resources that I wanted to share with you today. You know, as someone who comes from a family impacted by Huntington's disease, it's a rare neurological condition. I just want to thank CRN team who put on the conference and invited me to speak. You know, I know the volunteer hours and efforts that go into these conferences and have truly personally benefited from attending similar events throughout my many years. So thanks for the CRN team for putting this on, and really for all of you for attending. Hopefully it's been a good week, a good weekend, and hopefully the next few sessions are enjoyed as well. So if anything, I hope you walk away with a new resource today. And again, if you have any questions about anything about these resources or anything about Horizon, please don't hesitate to reach out to myself and my team using the email connect@horizontherapeutics.com. But appreciate your time today, and we'll pass it back over. Take care.
Thank you, BJ. That was excellent, great information. Thank you for sharing all the great resources that BJ that Horizon has to offer our community. As BJ mentioned, you know, we can. Any other questions that you might have, you can submit them to anyone on our board, to me via the info@Cystinosis email, and also through the channels that BJ had mentioned. So thanks so much for joining us and giving us all that great information. So we're going to move along to our next talk. We are so happy to have folks from Avrobio here to talk about the investigational lentiviral gene therapy for cystinosis. Fernanda Copeland, who is the Global Head of Patient Advocacy Engagement, is going to join us, as well as Jay Jones, who is an Associate Director and Clinical Scientist at Avrobio. We'll be providing information first, and then Fernanda and Jay will join us live at the end of the recorded presentation. We'll be able to answer questions to the best of their ability. I would like to note that there is a portion in your attendee hub where you can submit questions. It's the chat function. It's at the top right, and I think is probably very, very tiny. So if you're having trouble locating that, take a look at the top right of your screen. Please submit questions during the presentation and starting now, and we'll try to get them answered for you after their presentation. So here is our information from Avrobio.
Good morning, everybody. And thank you to the Cystinosis Research Network for having us today and allowing us to share our progress on our investigational lentiviral gene therapy program for cystinosis. I'm joined by my colleague, Dr. John Jeffrey Jones today, who will be giving an update on our progress for the program. I'm Fernanda Copeland, Head of Patient Advocacy and Engagement, and my responsibilities include the relationships with the patients and the patient advocacy groups to make sure that we are bringing to the development the voice of the patient. Before we begin, I'd like to remind everybody that lentiviral gene therapy for lysosomal disorders are still investigational, which means they have not been approved by the FDA or any regulatory agencies for physicians to prescribe to patients, and they are still being evaluated in clinical trials. So for today's brief presentation, I'll be sharing a little bit more information about the Agrobio Pipeline, our lentiviral gene therapy approach, and then Dr. Jones will give an update on the program. If this is the first time that you're hearing about Avrobio, we are a clinical stage lentiviral gene therapy company founded in 2015. Our focus is to develop personalized gene therapies for people living with lysosomal disorders. We're focused only on lysosomal storage disorders. There are 6 programs in which 3 of them are being evaluated in the clinical stage, which means in humans. There are about 140 employees at Avrobio, 2 offices, one in Cambridge, where our headquarters is, and one in Toronto. We are growing very fast, and we are really passionate about our groundbreaking science and our focus on the patients that we serve. This is the Agrobio Pipeline, Fabry being our leading program, where we have treated so far 12 patients in a phase 1 and a phase 2 clinical trial. We have data that goes far back as 4 years ago. We have treated in an investigator-sponsored trial partner with UCSD 3 patients in the cystinosis program, 1 patient in the Gaucher type 1 program. And the reason I bring up the number of patients that we have treated so far, so that's about 17, and that number is increasing very fast as we plan to also treat somebody for Gaucher, Hunter, and Pompe this year or next year, is because the more patients that we are able to treat, the more we know about the safety of the lentiviral gene therapy. So gene therapy in general is a very vast field that is a very generic and large term that can mean a lot of things. Gene addition, gene augmentation, gene editing. So what gene therapy really means is a technique that uses genetic material instead of surgery or a pill for the long-term treatment of a genetic disorder. Avrobio is working on monogenic disorders, which means one change in the gene or one variant gene. That's a monogenic disorders when there are more than one mutation, then that would be a polygenic disorders. We're focused on monogenic. And this transfer of therapeutic gene, genetic material is done by utilizing a vector. It's called a vehicle or a vector, and the picture that you see on the bottom is a vector. And we utilize a vector in order for the gene transfer to be added to the person's cells and to be integrated into the DNA. What you see is a lentiviral vector, which is a highly modified virus that is no longer able to reproduce or spread. Once this vector is inside the cell, then the capsid or the envelope that you see outside is dissolved, and then the expression cassette is able to start producing the protein that was missing. In the case of cystinosis, the protein cystinosin is starting to be produced by each therapeutic gene that is inserted into the patient's cell. So Avrobio is focused on ex vivo lentiviral gene therapy. The word ex vivo means outside the body. Lentiviral is the vector that I showed you in the previous slide, gene therapy. So the way that it works is that the patient would go to the clinic and receive a medication to mobilize peripheral blood so we can collect the stem cells that are needed for the gene therapy. In this first step, in the second step, the cells that are collected, the CD34 cells, also known as trunk cells, the trunk cells are kind of like generic cells that in the future they will divide and they will become specialized cells. with their own function. So we collect and select via a process called apheresis the CD34 cells. Once we have these cells, then these go to the manufacturer where they get integrated with the vector that I mentioned to you that has the therapeutic gene. That process is called transduction. That's when the cells of the patient and the vectors are integrated, and then so the therapeutic gene integrates into the cells and into the DNA of the patient. The goal is that every cell has a copy of the new working gene that was missing. Once transduced cells are present, then these go through a series of tests to ensure that we have the drug product that we want that is high quality. And then they get cryopreserved, also known as frozen, and shipped to the clinic where the patient will go through a process called conditioning to be able to receive the one-time infusion of gene therapy on step number 5. So remember that I mentioned that we are collecting CD34 cells, and these are trunk cells that divide into different cells. Once the gene therapy infusion happens, there is going to be a large population of short-term progenitor cells that are produced very fast after the gene therapy, and they will last for the first 6 months post-gene therapy. Also, from those CD34 cells, there's going to be a burst of long-term engrafting cells. But this is not as big of a burst, and it tends to happen after 6 months of the infusion. These long-term engrafting cells will continue to divide for the long term and hopefully for the lifetime of the patient. Every cell division that happens will contain a new working copy of the gene. And in order to get prepared to receive the gene therapy product, the patient must go through a process called conditioning. Conditioning is an important process in lentiviral gene therapy, and the purpose is to make space in the bone marrow to allow for the patient's new transduced cells occupy space in the bone marrow and be able to take hold or engraft and divide into future cells. Avrobio uses busulfan as a conditioning agent. Busulfan has been used for many years for a variety of uses, including as chemotherapy in certain types of cancer. We utilize busulfan for a period of 4 days, and the dose of busulfan that is used is individualized to each person because every person has a different metabolism. So every person's exposure to busulfan will be unique to its own individual. We measure every day to try to reach the exact amount that is needed for each person to maximize engraft, take hold, and minimize side effects. And now I'll turn it over to Dr. Jones, who will give an update on our program. Thanks, everybody.
Thank you, Fernanda. Next slide, please. I'd like to start here by describing the mechanism of disease and then explain how it relates to Avrobio's investigational therapy. Cystinosis is caused by non-functional cystinosin, a protein with a number of different functions, including transport. The protein cystinosin is embedded in the membrane of the lysosome, and the job is to transport cysteine out of the lysosome. This is depicted in the figure on the left with the protein seen along the perimeter of the lysosomal cell. The lysosome is responsible for digesting and recycling cellular materials. When this doesn't occur, cysteine levels build up to a level where cysteine turns into crystals, and then these crystals damage cells and their integrity, and it happens throughout the body. Our investigational therapy intends to provide the functional cystinosin protein to perform the multitude of functions, and we expect much greater systemic benefit and potentially including to the central nervous system. Next slide, please. As explained, in cystinosis patients, cells in the body lack functional cystinosin protein. And with our therapy, cells are designed to restore the normal cysteine recycling. So cystinosin, the protein, is transported by a variety of different mechanisms, including tunneling nanotubules and exosomes. In the picture below, on the left is a macrophage cell, which is a white blood cell of the immune system. And on the left, it's the macrophage with a corrected gene, which recognizes the defective cell on the right without functional cystinosin. Cells with the transgene can either directly connect to other cells by this nanotube. The name comes from the tube-like structure, and then it's a transport mechanism in the body, or release of exosomes, as you can see on the picture. And these act as transport vehicles of molecules that can be taken up by cells. It is believed that through these mechanisms described, that our therapy allows the corrected lysosomal cells to migrate from the bone marrow, where they engraft to other organs of the body, including the kidney and eye. Next slide, please. So cystinosis is regarded as a disease with high unmet medical needs. The following list here describes the aims of Avrobio's gene therapy approach in cystinosis, which includes improving renal or kidney function, improving your vision, endocrine disorders, central nervous system complications, and corresponding developmental issues, as well as, importantly, to lessen the everyday burden of the disease. Thank you. Due to these high unmet needs, there's an ongoing first-in-human phase 1/2 trial of our gene therapy conducted at the University of California, San Diego, with doctors Stephanie Shirky, Bruce Barshop, and Edward Ball. And in this trial so far, 3 patients have been dosed, which is very exciting. Turning your attention towards the bottom, the objectives are primarily to demonstrate safety and tolerability, followed by hypothesis generation of endpoints. And so measuring taking different assessments in different organs and looking at how the therapy responds. This study allows an enrollment of up to 6 patients, adults and adolescents, male or female, and those that are currently on cysteamine, and that includes both the oral systemic version as well as the topical eye drops. Next slide, please. All right. So this slide depicts the patient experience of going through the clinical trial and the order of events in the process. I'll start first by orienting you to the center of the graph, day 0, which is when patients receive the gene therapy and corrected cells, so that you can take note of this time sequence related to the procedures that I will describe on this graph. So scaling back in time to the left of the graph, in the beginning, patients go through what is known as a screening period. And this is about 8 weeks or so, in which patients will perform a series of tests to ensure that they are an appropriate fit for inclusion into the trial. If they are deemed appropriate, patients will then undergo baseline assessments, which will serve as the formal point in time which we will analyze relative to. If deemed appropriate, these patients will then undergo the pre-transplant procedures for a period of about 6 to 8 weeks, during which important steps are taken to ensure to prepare the body to receive the modified stem cells. A patient will receive, marching along left to right, a patient will then receive medications that allow stem cells to be released from the bone marrow during what we call mobilization, and specifically plerixafor and GCSF. That's a granulate colony stimulating factor agent. And again, this is performed in order to get those stem cells mobilized from the bone marrow. The next step is apheresis, and this is in which the procedures are to obtain the cells that we want to modify. And then there's a period of refinement and manufacturing that occurs in the interim while the patient prepares to undergo a personalized conditioning with precision busulfan dosing. And this is over a period of 4 days. And what this is it allows for the body to make space within the bone marrow compartment in which the modified stem cells are then expected to engraft. That key word that we keep talking about, which means take hold in the body. So the stem cells will divide and then from there. And then a washout period is necessary prior to the infusion and just after receiving the busulfan agent. And this is necessary over a period of a few days to then allow the busulfan to clear from the body, work through the metabolism processes and not damage the new cells to be received on the day of transplant. So then comes what we call day 0. And then following that, there will be, this is a larger grouping here, but you can see that there are many tick marks for patient follow-up. And then the patient will be expected to return to the clinic for these follow-up visits in which important measures of both safety, efficacy will be obtained, as well as just general regard for the well-being of the patient in the study. Over a longer period of 13 years, there will be continued follow-up to adhere to regulations. For all patients that receive gene therapy, the FDA requires a 15-year follow-up duration, and this is to further assess the safety, durability, and a number of other measures. Next slide, please. So the University of California, San Diego Phase 1/2 study will support safety and tolerability and give key insights into the clinical measures for future Avro clinical trial. And I'm very excited to be working on this project. Avrobio has partnered with Dr. Stephanie Shirky to help inform this pivotal trial that is being designed. We've listened to key opinion leaders, these clinical experts from all over the world, as well as the patients via advisory boards to help inform our program. And we're in the process of designing this trial and communicating with regulatory agencies to gain clearance to proceed further. And I'm excited to point to here that Avrobio has opened an IND, and it's as the sponsor of it. An IND application is an investigational new drug application, and this is permission, so to speak, from the agency to move forward with development plans. And as far as next steps go, we plan to initiate this global registration enabling study, which we just call the pivotal study. And this will have sites we're planning to have in the United States as well as Europe. I'll mind you that we're still in the early phases of planning and that we look forward to communicating our plans as they develop. Next slide. So on behalf of the Avrobio organization, me, Fernanda, and the many others that are all working on this as thanking the Cystinosis Research Network for organizing this forum and for allowing a great exchange of science and for allowing us to share our progress. Also, a big thanks to the patients and families for participation, because without participation of this, we wouldn't be as far along as we feel that we are. And so thank you so much. And if time permits, we can take any questions. So thank you so much.
Okay, thank you so much, our partners at Avrobio, for that great presentation. I'm joined live here now by Fernanda Copeland and Jay Jones, who have generously agreed to be here to answer questions, and we have a little bit of extra time because we've been so efficient this morning. So before we start in with that, I just want to mention that we've been very blessed to have Avrobio as such a great partner in what we're doing, and have been so transparent about their plans, have worked with our patient community, via us at CRN, via the Community Advisory Board internationally, and we think that's really the right way to do development in rare disease, and we're thankful that we have you as partners. So let's go into some questions. Fernanda and Jay, I don't know if you have any initial thoughts that you'd like to share, but I do have some questions directly from the community that are being inputted into the chat, and people can continue to do that as we go. But I'll start with our first one, and Amanda and Jay, you guys can decide who's better to address them. So our first question is, how can parents living in other countries involve their children in future trials?
I can take that, Jay. Thank you. That's a great question. And we realized that the first clinical trial, the phase 1/2, was in the United States, one site, which is can be typical for rare diseases at time. So the way that we have done participant identification for other clinical trials, and that we think it's a good way to do it, is we work, so all the stakeholders that work with us, and stakeholders are patient advocacy organizations, meaning the patient community, the regulatory agency, the clinicians working on this. We all share the information at the same time with everybody. So we plan to have a clinical trial website where everybody receives information on the clinical trial and the opening of the clinical trial at the same time. There is also a way to apply for the interest in the clinical trial, and there is a kind of a blinded process to go through it, and then clinicians who are working on the clinical trial would receive the interest of the participants. That's one way that we can do it. And back to your questions about international, one of the things that it's important for us to know is that the clinical trials will likely open in different countries at different times. So if it opens in the United States first, then possibly patients from the United States will be considered first, and then other countries. So that is something to know and consider. I hope it's helpful.
It is. Thanks, Fernanda. I have a related question, and this is sort of, it goes maybe to the same points, and maybe your response will be the same. But I've had a variety of questions about what the age will be for inclusion in the trials, and I don't know if you can speak to that or not. But
I can say that there's a lot of factors that we need to take into consideration for that. And I understand that treating earlier can mean preventing future complications. There's a lot of factors that we need to consider when deciding the age, inclusion, exclusion criteria. And I'm going to answer part of it, and then I'm going to turn it over to Dr. Jones because he's directly working on the clinical design of the study. But what we propose is one thing that we consider with the physicians and the patient communities. And then we also need to hear from regulators what they think it's safe at this point. I'll turn it over to Jay to add.
Thank you, Fernanda. Yeah, with respect to the question, it's we have been speaking with the experts in the field and really critically evaluating the ages in which we will seek to include based upon the design. I'll say that I've been working with Avrobio for a year now on the cystinosis program, and we've been advancing it very carefully and thoughtfully.
Also, just to add, so with every clinical trial, there are potential risks associated with it. And so when involving pediatric patients in a clinical trial, we need to consider those risks, potential risks as well.
Understood. Thank you, Fernanda and Jay, for those answers. I have another question, and this sort of goes to, and I'd like to highlight the difference between the trial that Dr. Cherki has been conducting, and then how development will move, into Avrobio and what they've shared with us today. But I do have a question on that initial trial and the patients that have been involved so far. And maybe you can answer or not, but you can let me know. The question is, does the pandemic slow down publishing of new data from the first 3 patients?
I think Jay is a better person to answer this question.
So with respect to the pandemic and the clinical trial and publishing of data, we cannot ignore the impact that COVID-19 has happened, and we had incorporated steps along the lines of the FDA guidance to really address and mitigate the effects. So on timing, I don't have a very clear sense of when we may be publishing that. However, we did seek to address and mitigate any impact from the COVID-19. So,
this question, and you guys had this question yesterday too about publishing. It's so far everything that is everything that is Dr. Cherki's data, she has been the first one to publish. Jay, this is correct, right? To your knowledge?
That is correct.
Yeah.
Okay.
I think she's presenting somewhere. She has been presenting in conferences. There are videos and updates that can be accessed online that has been posted. I know she'll be presenting somewhere again next year in March. And then the company Avrobio, when we release, again, back to our stakeholders, when we release data to the investors, they become available. And so we try to release the data to everybody. I am not aware of any manuscript right now, but maybe there is one that I'm just not aware. Yes, Jay.
And ultimately, I think the decision is Dr. Stephanie Cherki's and UCSD to publish that data.
Yes.
I think that's a good point to make, and it's a finer point of drug development, and it's difficult for the community to understand sometimes because we just see it as we're developing gene therapy for cystinosis, and folks are hungry for information and updates on it, and the difference between Dr. Cherki sharing her data, whether things are published or pushed out to the community. But I can assure our community here that's joining us today that Avrobio has been very transparent, and Dr. Cherki, and we've been able to share whatever is public and available. We've made an effort to share it, so we appreciate that. So we have a few minutes left. Being a little ahead of schedule is a good thing. It allows us a little bit more time for questions. So I have a couple of specific questions about the conditioning regimen in the trial. And I don't know if you can share a little bit more. So I think sometimes it's difficult for people to understand. Conditioning is a new word, a new terminology for us in our community. And and people see that the drug that's used for the conditioning process is a chemotherapeutic agent. And maybe you can go into, and Jay, maybe this is for you, but go into explain a little bit more fully about, what that process is like, why you use that agent, and how that might feel for someone participating in the trial.
Absolutely. I'm a pharmacist by training, and then so keenly aware of the medication and being able to explain this to the audience here. So busulfan is historically a chemotherapeutic agent, and this it's an agent which depletes other cells within the body, and it's very important in order for our therapy to work and be successful is to administer this medication so that within the bone marrow space, there's a depletion of cells so that when we put the stem cells into that compartment, there will be enough space and then they can grow and divide. A little bit more about our approach with busulfan is that it's through a therapeutic dose monitoring, and this measures the concentrations of the drug in the body and as it is metabolized, and there's a targeted amount that we are looking to achieve within the patient. So based on extensive literature and analyses that we've determined to be the sweet spot, so to speak, between just efficacious enough without causing undue or excessive side effects. And so that's the goal, really, to strike a good balance with administration of busulfan and to prepare the body. It's a critical step, and the concern is definitely understood, but we've safety. We've gathered evidence and throughout our programs of just the tolerability of this method, and so it's if I can dispel concerns, I hope that did.
I think that is helpful, Jay. I appreciate it. I think that's one concern that folks have had, but again, I really appreciate all the extra information you've been able to share with us today. The recording was really helpful. Fernanda was able to join us for part of our medical panel to answer a few questions yesterday, and that was also really helpful to get information out. So with that, I think we'll wrap up our Q&A portion of the Avrobio talk. Again, thank you so much to Fernanda and Jay for joining us today and for providing such great information. And if you have more questions that come up, please send them to me via our address at info@cystinosis.org, and I'll make sure that I get them to our partners at Avrobio. So next, we'll go to our next speaker, and at this point, we move to updates from our CRN funded investigators. We're happy to highlight their work that they're doing in the cystinosis research world, lots of different topics. And we're going to start now with a presentation from Dr. Caterina Hohenfelner, who is a cystinosis physician in Germany, and also Harold Schubert, who is from the company Novotech. There'll be a series, and one of their colleagues will present as well. There'll be a series of recorded presentations, one about the disease-specific quality of life questionnaires that their team is working on, one on IMPACT, which is the improvement of motoric abilities in patients with cystinosis, and then finally an update on their newborn screening project. So with that, I will move to our presentation from Dr. Hohenfelner and her team.
Good morning and good evening, dear patients, dear families, dear organizers. First of all, I would like to thank the organizers for this meeting and the opportunity to present our projects and data. I will give a short update on the pilot project on newborn screening for cystinosis and the two projects that are now planned in regard to the screening. In the second part of our talk, we will talk about the improvement of motoric abilities in patients with cystinosis. First, Susie Bechtold-Dalla Pozza will report on PQ-CT measurements in patients with cystinosis. Then Harald Schubert will give an introduction to the Galileo plate, which we used in our impact study. And I will present some preliminary results of the study. So far, 332,000 newborns have been examined in the pilot project for cystinosis screening. 3 patients with cystinosis have been identified. This corresponds to an incidence of approximately 1 in 100,000 newborns. In order to implement cystinosis screening into normal newborn screening in Germany, we will carry out 2 further projects. The first one is an evidence-based guideline for cystinosis. We are currently writing the grant proposal for this project, and the project is planned to be carried out with the International Cystinosis Group and Patient Support Group. The second project is a project that has been carried out by Julia Quittmann. In this project, quality of life questionnaires will be developed, especially for cystinosis patients. The project will start in October 2021. Julia Quittmann will invite you to an online presentation in early fall. And now I would like to hand over to Susanne Bechtold.
Patients, dear families, I had the opportunity to measure in about 50 patients with nephrogenic cystinosis bone mineral density, and we used the peripheral quantitative computed tomography. As you can see here, this is the machine, the front and the side view. And what can we measure? We measure bone mineral density at the peripheral forearm and the proximal forearm. And what do we get? We got the trabecular bone mineral density, which is situated inside the bone, and from that we can measure the whole bone mineral density at the peripheral skeletal localization, and also the trabecular bone. And at the more proximal side, we measure the cortical bone, which is here in dark gray, and the surrounding muscle area, which puts the forces on the bone, and the bone is driven by the muscle. It's very important. And from that, we can also have a look on the bone geometry, which means how the cortical bone situated within the forearm, and we can measure the total bone area and also have a look on the inside, how large is the medullary cavity and how small or large is the cortical bone area. And all these measurements were done in patients with nephropathic cystinosis. And this is an example of a patient measurement. And here you can see it's in red, it's the trabecular bone. In light gray is the muscle area, and dark gray is the fat area. And as you can easily see, we can also have a look on the geometry of the bone, especially in the peripheral, on the proximal side of the measurement. And we investigated within the study situation 41 patients within 17 females. They were children and young adults. At the whole group, they were shorter and lighter in comparison to a healthy reference group, and the trabecular bone and the cortical bone mineral density were normal. And you can see here, this is a normal trabecular bone density, and this is reduced in osteoporosis. Though these patients have no osteoporosis, they have a normal cortical bone density and a normal trabecular bone density. What do they have is an abnormal bone geometry, which comes by the lower muscle mass and especially by the reduced muscle function, and this we measured by grip strength and jump force. And let me illustrate the altered bone geometry. So this is a normal, healthy control. And with patients with a nephropathic cystinosis, the medullary cavity, the hole within the bone is a lot larger. The total bone area is a little bit smaller in comparison to a healthy control. And as you can easily see, the cortical thickness or the cortical area is, in comparison to a healthy control, much smaller. And since the muscle drives the bone, and the patients have a lower muscle mass and function and a less coordination, we have got an altered bone geometry, and this increases the possibility that the bone will fracture with this small cortical area. The bone strength is reduced, and this depends on how far away the cortical bone area is from the center. This might be a little bit complicated, but you see there's less bone stability, and what can we do against it? We can increase the muscle function and especially the force, and this is what we have to do to increase bone health. We have to increase muscle mass, muscle strength, and coordination. Thank you very much.
Before I start my presentation, I give you some information about our background. We are since 30 years in the field of bone and muscle, and we are producing these small computer tomographs where we can quantify the bone-muscle relationship. Then we are producing some measuring devices where we can quantify daily movements like sit-to-stand, jumping, balance in physical units. And then we have specific training devices based on mechanostimulation, like tilt table for people who are not able to stand, dumbbells for the hand and arms, and standing devices for people who are able to stand. My talk today is about mechanical stimulation. That means stimulation of the neuromuscular system in a physiological way. And my main topics are what you need for mobility and independency, and the physiological effects and benefits of mechanical stimulation. Coming to the first topic, so mobility is related to muscle power. And often in medicine, they're just looking to muscle force. But for instance, somebody who has spasticity can generate a high muscle force, but he's not able to make any movement. So he has no movement velocity. So muscle force times movement velocity means muscle power. And during our daily activities, we have two types of movements. We have high power movement for a short period of time, like chair rising, stair climbing, jumping, or we have low power movements over a long period of time, like cycling and swimming. If we look now to the typical power of a person with around 80 kilogram or 176 pounds, so on a bicycle ergometer, this person is doing in average about 80 watts. Sit to stand means already 10 times more than cycling, 800 watts. And this is the same power which you need normally for stair climbing. And if we talk about jumping, it's even 40 times more than biking. And jumping is normally also very important if we talk about fall prevention. Often people with a metabolic or genetic problem are not able to resist an exercise or high muscle power training. And often they're just doing cycling, swimming, or walking, but this means no training of high muscle power, no training of energy storage, limitation by the cardiovascular system, depending on the motivation from the patient, and needs a minimum of functionality, so difficult for a disabled patient. And now with mechanostimulation, you have now a method where you can train the high muscle power, but without having the problem with high impact on the cardiovascular system, for instance, and other benefits. Now we are coming to mechanostimulation, and here you see different devices. So we have tilt tables, smaller ones and big ones. For people who are not able to stand, we have standing devices, and for the upper extremity, we have a dumbbell to train arm and hands. Looking to the application of Galileo training in medicine, then we have studies in osteoporosis, back pain, geriatric, urology, neurology like spinal cord injury, stroke, MS, Parkinson, decreased mobility with lung disease like COPD, decreased mobility and pain reduction with chemotherapy in oncology, children with neuromuscular diseases like CP, SMA, spina bifida, Duchenne, OI, osteogenesis imperfecta, and sport injuries like ACL, tendon, range of motion. In this graph, we give you an overview about publication between 1998 and 2019 in the field of mechanostimulation. And you can see that we have meanwhile more than 377 Galileo publications between 1998 and 2019. The first very important thing to understand is that there are basically only two options to stimulate the neuromuscular system from the outside. So you have electrostimulation or mechanical stimulation. And people know electrostimulation, but they are not so familiar with mechanostimulation. Everybody knows the patellar tendon reflex. That means we give a mechanical disturbance and you get a muscle contractions. If we look now more in detail what happens in the body, so we hit with the hammer here on the patellar tendon, and then the muscle spindle is elongated. The muscle spindle sends a signal to the spinal cord, activates the muscle. So if we compare this now again with electrostimulation, in electrostimulation you are stimulating the afferent nerve, and so you can create a strong muscle or a big muscle, but not a very functional muscle. With mechanostimulation now you have the neurology, that means the afferent and efferent pathway, plus the muscle. So we see the Galileo training as an additional tool in rehabilitation to make rehabilitation more efficient, and you can do something which you can't do with standard therapy. So what you are normally doing is endurance training like cycling and swimming, resistance exercise like weightlifting, resistance training machines, coordination and balance, or functional training like sit-to-stands, walking, stair climbing, or treadmill. And now with the Galileo training system, you get an additional tool where you can do something which you can't do with the conventional methods. And the first thing is that you can stimulate the neuromuscular system from the outside. You can train the high muscle power, but only applying small loads, so it's very safe. The influence on the cardiovascular system is quite small and very well tolerated even in patients with heart and lung diseases. And very important if we talk about muscle function, and muscle function means that we need a lot of repetitions. And normally with conventional training you have to make maybe 2 repetitions or 5 repetitions, but depending on the frequency, here with the Galileo training you can go up to 30-36 repetitions per second. The basic principle of Galileo is that we're making this side alternating movement. So we are tilting the pelvis like in walking, that we can activate not only one muscle group, but a complete muscle chain. That means the trunk muscles, the pelvis muscle, and the leg muscle as a complete muscle chain. But it's like in walking, but without swinging phase. The stimulation effect is like we have seen before. So we elongate the muscle spindle. The muscle spindle sends a signal to the spinal cord and activate the muscle. And independence of the body position, so if you have more straight legs, it's more going to the upper body. If you're more flexed legs, it's more the hip, pelvis, and leg muscles. And that you can really measure the stimulation effect of mechanostimulation. This is now a video from the sport fields in the US. So they have put 2 EMG electrodes, and here you can see the 2 signals of the 2 EMG electrodes. And first they start with a squatting exercise without Galileo. So you can see here now the 2 signals without using the Galileo. And here at the end they are now switching on the Galileo, and in the moment the machine starts to work. You can see now the much higher muscle activation by using this mechanostimulation device. Now we are coming to a very important topic, how we have chosen the frequencies and which frequencies for which application. And the first consideration comes from the muscle side. The muscle needs about 25 milliseconds to contract and about 25 milliseconds to relax, in total 50 milliseconds. And if you divide 1 second by 50 milliseconds, you end up by 20 Hertz. This means if the frequency is below 20 Hertz, then the muscle can still contract and relax. If you go above 20 Hertz, the relaxation time is cut off and you get a more and more higher muscle tone. The second consideration comes from the neurological side. So if you elongate the muscle spindle here and the signal runs up to the spinal cord and down again, this loop takes also about 50 milliseconds, and this corresponds also to 20 Hertz. And the third consideration comes from the central feedback system. That means the communication time between the foot and the brain, and this needs about 80 to 100 milliseconds. And if you divide 1 second by 80 to 100 milliseconds, then you end up by 10 to 12 Hertz. This means if the frequency is below 12 Hertz, then our brain has to compensate the side alternating movement, and we have not anymore the spinal feedback system. If we look now to the frequency range and the therapeutic application, so the low frequencies we are using for balance, mobilization, and proprioception. The middle frequencies, where the muscle can still contract and relax, we're using for stretching muscle, muscle function, or coordination. And the high frequencies, above 20 Hertz, we are using to build up muscle power, build up muscle strength, and also, if necessary, to reduce spasticity. So the frequency determines how you want to targeting the muscle. So this is the first parameter to choose normally. The second parameter is then the foot position. That means if you are very close together, you have a low intensity, and if you go wider, then you have a high intensity by the same frequency. And already simple voluntary exercises in combination with Galileo provides an overall neuromuscular training. Here you can see some basic position how to train. So you can do a squatting exercise, you can doing sitting exercises, or with the arms on the machine to train shoulder, neck, and arm muscles. The next benefit with Galileo training is that we can train the high muscle power, but only having a low cardiovascular effect. So even deconditioned patient doesn't get exhausted by using Galileo, in opposite, for instance, to resistant exercise. And the reason for that is that the muscle can work in 3 different functions. So the first function is that the muscle has to generate energy, and this he has to do if you're standing up from a chair, stair climbing, cycling or swimming. Here the muscle has to generate energy. The second function is that the muscle works like a spring, so he stores the energy and releasing the energy, and this happens, for instance, in the free selected walking speed is the speed where you can move your body over hours without getting exhausted. And the last function is that the muscle works like a shock absorber. He has to convert the kinetic energy in heat, and this happens if you're walking downstairs or walking downhill. And here you get much easier muscle pain than walking uphill. And if we now compare standard therapy with Galileo therapy, in standard therapy the muscle has always to generate energy. In the Galileo training or on the Galileo training, here the muscle works only as a spring, and so the impact on the cardiovascular system is very small. The next very important benefit is that the Galileo therapy is very safe. That means the loads in the joints and ligaments are less than walking. In this graph now you can see the loads during daily movement in comparison to the loads during Galileo training. And for instance, if you do one leg hopping, you're doing about 3 times your body weight. Already in walking, you put more than 1 time your body weight on one leg. And if you compare this now with the loads during Galileo training, the loads are less than any daily activity. So the risk to damage joints, ligaments, bones, muscles is very, very small. And in the last 20 years, there was never have been a severe side effect. And here you can see now an application in children with osteogenesis imperfecta. So even in children with brittle bone disease, they can do training at home on a tilt table without having the risk to get injured or getting fractures. And they are doing this training twice a day at home to train the neuromuscular system. And then she has learned to sit and standing. In this video, I have only now the sitting. Another benefit with Galileo therapy is that you can get this high repetition rate, which is necessary to improve muscle function. That means better intramuscular coordination and a high neurological input to the brain. And if we look in the literature, what is necessary to learn or relearn a movement. So in motor learning, we know that to learn or relearn a movement, you need 100 or 1,000 of millions of repetition. So for instance, learning to walk means about 3 million steps, or if you are a professional baseball player or something, you need more than 1.6 million throws. So if you talk about learning something, we need a high repetition rate. And now if you look to the number of repetitions with Galileo, so for instance, if you are training with Galileo at a frequency of about 25 Hertz, it means 25 muscle contraction per second, 1,500 muscle contraction per minute, or 4,500 muscle contraction in 3 minutes. So Galileo is probably the only tool where you get this high number of repetitions. Coming again back to the general indication of Galileo, so training of muscle power and muscle force after immobilization and injuries, prevention for osteoporosis, balance training and prevention of falls, coordination training, muscle atrophy, back pain, bad blood circulation, pelvis muscle floor training, stress incontinency therapy, decrease of muscle origin and or muscle contractions. Maybe more important, the list of contraindications when not to use the Galileo. So pregnancy only as a precaution. We don't see a sense to train women which are pregnant. One which is very important in the moment is our artificial joints in the leg and hip, because we have studies who shows that the loads in the joints are very small. So normally quite safe to use it even with patients with artificial joints, but we know that 1-2% of these artificial joints can be lost by something else. And in the moment we have no large study, and therefore it's in the moment a contraindication. On the other side, plates, screws, pacemakers, baclofen pump, stents, shunts are no contraindication. Open wounds, because we don't want to cause stress on the wound. Recent fractures, you wait normally 10 to 12 weeks. The second most important one is if there's a risk of acute thrombosis. This is not only related to Galileo training, but to any training. The same for kidney gallstones. Patient with epilepsy, only because of the forest, there's no sign that we have a negative effect on epilepsy or that we provoke epilepsy. Bone tumors because of the reduced bone strength. And strokes where still is a risk of another stroke, but afterwards it's a perfect tool to train. Here you see now special devices we have for the arm and hands. So here's a dumbbell for the adults or 2 types of dumbbells for kids. And possible benefits which we see for patients with cystinosis. So stimulation of the neuromuscular system in a physiological way, improved postural control, gait and walking speed, improved muscle strength, mobility and coordination and function. An indirect effect is on the bone. By creating higher muscle forces, you can increase the bone mass, muscle mass, density and osteoporosis prevention. Increase joint position sense, balance and proprioception, intermuscular coordination, neurological communication, optimize plasticity, improves blood flow, microcirculation in the lymphatic system. Thank you very much.
Now I would like to give you a short update on our study impact, improvement of motoric abilities in patients with cystinosis. Primary objective of the study were: does regular home practice using Galileo vibration training platforms positively impacts muscle strength and increases cardiorespiratory performance in cystinosis patients? Secondary objectives were: does the training result in a general increase in everyday activity and an improvement in the quality of life? And is it possible to integrate the training into the day-to-day life of cystinosis patients? As study design, we used a matched pair design. The patients have been prospective randomized in 2 different groups. One group was training with vibration platforms, and the other half of the patients are training with vibration dumbbells. We included 40 patients in the study, 24 adult patients and 16 children. Inclusion criteria were confirmed diagnosis of infantile nephropathic cystinosis, and exclusion criteria a recent kidney transplant or a recent orthopedic surgery. The patients are undergoing 4 examinations at the beginning of the study, after the training, 3 months later, and 1 year after the study started. They have a physiotherapeutic and an orthopedic examination. The impact of the training is measured by different tests. As example, with a Leonardo plate, with a balance test, the grip strength, a single 2-leg jump, and we have additional tests as the 6-minute walking tests. Here you see on the left-hand side exercise with the Galileo plate. This is a squat, a 2-legged squat, and on the right-hand side, the training with the dumbbells. The training schedule in this study was 14 weeks, 7 days a week, 10 training units, and one training unit was 5 to 8 minutes long. The documentation was daily, and there was also a supervision included, an online supervision, and this was every 2 weeks or as often as needed from the patient. Here are preliminary results. In the group of patients who used the Galileo plate, it was shown in the heel rise test and in the 2 single leg jump that they were able to maintain or improve their performance despite COVID and their reduced general activity. The same could be shown for the training with a dumbbell with the right hand. In September, the adult patient will have their last examination, and at the same time, the study for the children will start. Due to COVID, we had to separate the groups. So the study will be completed in September 2022. This is the group who is involved in the study, and the main person behind the study is Christian Köppel. Thanks to the support of Cystinosis Research Network, we could perform the cystinosis screening and impact. Impact was also supported by the Irish Patient Support Group, the German Cystinosis Patient Support Group, and the Cystinosis Foundation Germany. Thank you very much.
Thank you so much to Dr. Hohenfelner and her team for that excellent presentation. CRN is proud to support the variety of work that Dr. Hohenfelner is involved with, in particular, the work on the impact study and newborn screening. There were several questions during the medical panel yesterday, and so we're hoping that her presentation and Harold Schubert's presentation address those questions. So now we'd like to move to our next presentation from another CRN-funded investigator. We're going to get an update on cognitive control systems and cystinosis from Dr. Ana Alves Francisco. She is a cognitive neuroscientist, working at the Albert Einstein College of Medicine in the Bronx, New York. She works with Dr. Sophie Malholm and Dr. John Fox, and their group has been involved in the cystinosis community for quite a long time. We're proud to support them. So I also want to mention that you can submit questions into that chat function. I think it's small and a little difficult to find, but it's up in the right corner of your viewing screen. So please submit those questions. We'll have some time at the end of Dr. Francisco's presentation for some live Q&A. So with that, we'll view Dr. Francisco's presentation.
Hi, I'm Ana Francisco, and today I'll be talking to you about cognitive control in cystinosis. I'll be the talker today, but I'm not alone, so I'd like to acknowledge the instrumental roles of Dr. Sophie Malholm and John Fox, Dao Ostes, and Elena Brutti on our cystinosis projects and on the work I'll be presenting today. I'd also like to stress that I'll be happy to answer any questions you may have about these findings and our work in general. So feel free to contact me. My email is here and will again be on the last slide. All right, so just a quick overview of what we'll be covering today. We'll start with the background. So I'll tell you about cognitive control and focus on two specific components of cognitive control: response inhibition and error monitoring. I'll then characterize our participants and describe the task we use to measure those cognitive control components. I'll go ahead and walk you through our findings. I'll introduce some strategies that can be useful in training cognitive control functions. And lastly, I'll talk about our new and exciting project. Okay, background. So let's start by defining cognitive control. So cognitive control is the ability to engage in goal-oriented behaviors, allowing the brain to solve difficult, novel, or complex tasks, such as correcting errors or overcoming automatic responses in favor of task-relevant responses. So as I'm sure you could guess by that definition, cognitive control is many things. Today, we'll focus on two: response inhibition and error monitoring. Okay, so let's start by defining response inhibition. So response inhibition is this ability that allows us to inhibit our impulses and natural, habitual, or dominant behavioral responses to stimuli, which in science we call prepotent responses. And we do this in order to select a more appropriate behavior that is consistent with our goal. And stopping yourself may seem simple, but really being able to inhibit a response requires fast adjustments to your actions. And inhibition is important for many things. For example, controlling our behavior, which has to do with self-control and discipline, controlling our attention, and even controlling our emotions so as not to act inappropriately. And this relates to self-regulation. So we are going to think of response inhibition as the just say no to the brownie skill. In the lab, we can test response inhibition using tasks like the one I'll be showing you now. So go ahead and say the colors. Do not read the words. I will give you a couple of seconds. Not straightforward, right? So to perform the task correctly, our brain needed to actively inhibit something we do so automatically, which is reading. But really, response inhibition relates to a wide range of real-life skills. For example, imagine you are about to chase a basketball into the street and you suddenly hear the honking of a car horn. Not being able to stop in such a situation would have serious consequences. Other situations requiring response inhibition, now home and school work centered, are, for instance, raising one's hand before answering a question in class, ignoring distractions while working, reading the directions before starting an assignment or a new task, and waiting for one's turn to play in a game or to speak during the conversation. What about error monitoring? So when an error is made, detecting that error helps us to adapt our behavior and adjust our attention appropriately so as to avoid further errors. And this process seems to be related to stuff like motivation, but also to how we respond to our own performance. We'll be thinking about error monitoring as it shouldn't have eaten that brownie realization. Moving on from the brownie. If we are not able to recognize when we make mistakes, we may have difficulties, not only in academic and professional domains, as we fail to understand when we are not performing the tasks well, but also in the social domain, perhaps repeating actions others find irritating. So good response monitoring influences our ability to function adaptively in school and at work, environments in which we are frequently being asked to learn and perform new skills. Moving on to our participants and the task we use to measure response inhibition and error monitoring. So we tested 35 people with cystinosis, and as you can see by the age range, they were children, adolescents, and adults. We also tested 45 age-matched controls. These two groups responded to a go-no-go task while their brain activity was being measured. As the name indicates, in a go-no-go task, sometimes an action is required, in this case, a button press, and at other times, the stopping of that action is what is required. So let's see a more concrete example. So in this case, the participant should press a button for every picture that shows up on the screen. So a picture shows up, we press the button, and we call this a hit. The next picture shows up, we press the button, and again, we have a hit. But the person should not press if the picture is the same as the one before. So the picture shows up, it is the same, just the same as the one before. We should not press. If we don't press, we make what we call a correct rejection. But if we make a mistake and we end up pressing the button when we shouldn't have, then we call this a false alarm. Now, this task is actually harder than it looks because 80% of the pictures are not repeated, which means that we get very used to press the button, and that makes it harder to suddenly have to stop ourselves from pressing. And 2, the task is very quick, which means that it's easier to make mistakes because we don't have a lot of time to think. So you can see how this task will allow for 2 things. First, to measure how good our brain is at inhibiting a response that is kind of automatic. And second, to see how the brain reacts when an error is made. And that's what we are going to look at now. So first we'll focus on behavior, meaning how good were the participants pressing the button and stopping themselves from pressing when they needed to, and how long did it take them to press that button. So before we start comparing the groups, let me quickly explain these graphs. So you will see the groups in different colors, controls in blue and cystinosis in green. On the left side, you have the proportion of hits in this case. Regarding these weird shapes, all you have to know is that the wider the shape, the more participants scored at that level. Now, each of the circles represents 1 participant, with the exception of the dark gray circle, which indicates the mean of the group. So looking at the hits, we can see that although the distributions are similar, that is, individuals from both groups scored pretty high and a little lower. If we look at the dark gray circle, we see that those with cystinosis pressed in average fewer times than the controls. Now, looking at the false alarms, I remember that the false alarms represent times when the person pressed, but they shouldn't have pressed. There is no difference between the groups. So people with cystinosis performed similarly to controls. However, you can see that there are some people with cystinosis who had a particularly harder time inhibiting their responses. So they made more false alarms. What about time? And you can look at these graphs the same way as you did before. So regarding time, people with cystinosis took a little longer to press the button, particularly for the hits. Let's move on to the brain responses. Before we do, however, I'd like to take a moment to explain you what the wiggles we'll be looking at are about. So we use EEG to measure electrical brain activity. We wear these caps that look like swimming caps with a bunch of wires coming from it. Those wires have these little metal endings that, with the help of some gel, are able to capture our brain activity. And you can certainly see by our faces how much fun this all is. And I assure you that that joy has nothing to do with the lollipops. Anyway, the data that we record are, as I was saying before, a bunch of wiggles, as you can see here. So we need to average many brain responses to different things to hits, correct rejections, and false alarms to be able to make sense of the data. When we do that averaging, we'll end up with something like this. And here you can see time on the bottom and amplitude, which means how big the brain response is, on the left. And you'll notice all these different letters and numbers, and those indicate brain components or responses that are very well studied and that we know have specific functions or represent specific cognitive processes. For example, to study response inhibition, people usually look at the component P3, which you can see up there. We know that P3 reflects response inhibition because when we compare the brain response to correct rejections with the brain response to hits, the response to correct rejections is much bigger, as you can see here. So we usually say that the bigger that difference, the better someone is at inhibiting. What we'll be looking at is exactly that difference. And when we do, and here you can see again cystinosis in green and controls in blue, we see that the groups are very similar. So people with cystinosis seem to be inhibiting as well as the controls are. To investigate error monitoring, we look at the different components. We call it PE. And PE is thought to reflect error awareness. From the literature, we know that when we make a mistake, in this case, the false alarm, right? We press the button for a repeated picture. We see this large brain response that starts almost immediately after that mistake was done. So let's see how our two groups reacted to the errors. Again, controls in blue and cystinosis in green. PE is there for both groups, right? We can see this big brain response after the error was made. So both groups are aware of their errors. They are both monitoring their performance. However, here we see that people with cystinosis show a smaller brain response to their errors. And this can be explained by different things, and we'll keep investigating these results. But a smaller PE has been related, for example, to high levels of anxiety and differences in keeping attention and focus. So those could be some of the reasons explaining this difference here between controls and people with cystinosis. So to summarize, while response inhibition seems to function very well in cystinosis, there seems to be some actual differences in the monitoring system. And for some individuals, those difficulties seem to be quite drastic, actually. So this is one of the things that we are very interested in and that we will continue to investigate in more depth in the new project I'll introduce later. But let's change gears for a moment and discuss some things that can be done to address cognitive control difficulties. And let me start by saying that the programs that will most successfully improve cognitive control are those that require and directly challenge cognitive control functions and support them indirectly by reducing stress or improving the ability to handle that stress. That being said, there are generally 5 domains one can focus on. Goal setting, what do I need to accomplish? Self-awareness, recognize one's stronger and weaker abilities. So how easy or difficult will it be to accomplish the goal? Organization and planning. What are the steps that need to be taken and in what order? And how long will it take me to complete each of those steps? Flexibility and strategy use. Identify possible obstacles, the solution for those obstacles, develop flexibility in pursuing alternative courses of action when the initial plan fails or needs to be changed. And finally, monitor and summarize. So assess progress, review goal, review plan, what was accomplished, what did work, what did not work, why was that the case, and can we be more effective and efficient in the future? And if so, how? In a more simplified and concrete way, we could train cognitive control by following these steps. So stop and think, identify the solution. What do I want to do? Is there a problem with what I want to do? If there is a problem, what are the possible solutions? We'll consider the consequences to each solution, choose the best solution, and then evaluate the results. At school and at work, other specific steps can be taken to improve function. For example, provide a quiet, stable learning work environment, make behavioral expectations, rules, and regulations very clear, work on alternative ways to engage with errors, developing self-correction in a more autonomous, process-oriented, and constructive way, and finally, develop personalized checklists in the form of acronyms for most common errors, for example, for writing or math errors. For instance, the STOPS checklist reminds students to check sentence structure, tenses, organization, punctuation, and spelling. And regardless of your age, a great strategy to improve cognitive control and mood really is the freeze game. So we dance when the music plays and freeze when it stops. And then we dance quickly for fast tempo songs, slowly for slow tempo songs, but then we reverse the cues. So fast music will equal slow dancing and slow music, fast dancing. Lastly, I want to talk about our new project, which we are about to start. And the goal of this new project is to thoroughly characterize cognitive control in cystinosis, strengths and weaknesses, focusing on different components and skills. We're particularly excited about this one because it was developed after conversations with families and people with cystinosis who feel like so many of their difficulties are cognitive control difficulties. So as for this study, we'll have a group of people with cystinosis and a group of age-matched controls. For this new project, we are also adding a group of parents and age-matched controls. And this will allow us to better test what the impact of the mutation versus the impact of the disease is on the cognitive profile associated with cystinosis. Also, the parents' inclusion might or might not have been a request from our participants with cystinosis who felt the parents ought to do some work too. As I mentioned before, this project is focused on cognitive control systems. So we will use EEG to measure brain responses in different tasks focused on different cognitive control components. So memory updating, set shifting, conflict monitoring, and response inhibition. So we'll keep exploring response inhibition and that specific difference in error monitoring between people with cystinosis and controls. We'll further characterize cognitive control and cognitive function in general by using questionnaires and standardized tests measuring verbal, non-verbal IQ and memory, executive functioning, and adaptive behavior. Additionally, we'll consider sleep quality and how that influences our findings, age at beginning of treatment, cysteine dosage, transplant, was there a transplant, how many transplants, and GFR. And we'll relate all these measures to get at the bigger picture and hopefully get at those difficulties felt by people with cystinosis and their families and contribute to the development of targeted interventions, leveraging strengths and working on weaknesses. As always, your help and participation is the most important piece of the story. So we'll be reaching out to those of you who participated in our previous projects and will advertise on social media. But feel free to contact me if you're interested in participating or have any questions about the project. So you have my email, you have my office number, and follow us on Facebook for updates on our research. This was it. Thank you so much for your attention and hope to see you soon.
Thank you, Dr. Francisco, for that wonderful presentation. We're really happy to support your work in your lab with Dr. Fox and Dr. Malholm, and you're doing really interesting work on cognitive issues in cystinosis. So I want to welcome you live to our meeting today. We have a few minutes left in this session, and so I wanted to open this up to questions. If you haven't already, please feel free to submit questions into the chat, and we'll try to get them answered here. So Anna, one question that I have is, so these findings are really interesting. They build upon some of the work that Dr. Doris Trauner has done and her team, over the past couple of decades. And I'm just wondering, our caregivers and parents and individuals living with cystinosis often wonder, how can I take this information and these learnings and apply them directly to the day-to-day issues that I'm having or that my child is having? And I don't know if you can, if you're ready to make those kinds of connections, or if you can kind of comment on what people may be able to do.
Yeah, thank you, Christy, and thank you for having me. It's always a pleasure to talk to the cystinosis community. So I think the strategies that I give in the last slides, I think, of course, they are very general, right? Because we're just trying to fit everyone in the same box, and we know that that is not the case, of course. But I think going from those suggestions, so a lot of it has to do with avoiding distractors in the environment. And that is true for everyone. So someone at school, someone at work. So I think that's a big initial step that can be taken, but also small things that can train cognitive control. And those are those like thinking steps that one can train to just stop oneself before acting. Of course, it's always possible to go more individualized. So if there's a family out there that would like to reach out to me and say, look, these are the exact problems that we are facing, and maybe they don't fit exactly the overall strategies that you are giving, I'm happy to consult.
I really appreciate that, Anna. I think that's a great service to offer, and I think our understanding of the basis of these issues is really going to be helpful going forward. And like I said, CRN is really proud to support your work. So I wonder, another question is there a plan at some point, as we learn more and more based on the work that you're doing, to come up with some types of, and I know that you talked about, there are specific supports and solutions for specific individuals, but there's sort of general guidelines too. Are you, I know that you've started to publish, some of your work, in particular in regards to cystinosis, but I know you've also done work with other disease areas. And I wonder if there's any plan to kind of make, maybe not a handbook, but some kind of a publication or some kind of a guideline that folks could share with their teachers, their other support systems in school districts and beyond.
Yeah, I mean, I think that's the ultimate goal, is to be able to translate these findings that are not always easy to understand and apply in practice, and so to translate them into meaningful ways so that it's easier for a family to go to the teachers, to go to clinicians and say, look, this is what we should be focusing on. So yes, definitely. And I think we still need to learn some things, and I think with this new project and adding the parents to the story and really starting to understand the mechanisms behind these processes, I think after that will be the next goal for sure.
Well, I think that'll be really fantastic and a great addition, to the resources that we have available for those living with cystinosis. So unless you have anything else you'd like to add for our community, I think we've addressed all the questions that have come in. I really appreciate your time here and your excellent presentation. So anything else to add, Anna?
No, just saying thank you, and please reach out to me if you have any questions. I'm always happy to be in contact with the families and help where I can. So, and please be excited about our new project and come and visit New York and me, and we'll have some fun. So yeah, that's it.
Well, thanks, Anna. I think Anna gave her contact information. You can always contact Anna through me, through CRN, through the info@cystinosis.org email, lots of different ways. And we'd love for more people to participate in the great work that they do. So at this point, we are going to go into our break. That will go until the top of the hour, so 1 p.m. Eastern time here in the US. During that time, you can remain logged in here to the sessions. Turn off, you can go ahead and take a break, take lunch. You don't need to log off and log on. You can stay here. We'll come back at 1 o'clock Eastern time, and we'll hear from Dr. Elena Levchenko. She'll give us information on her new validation study of a new biomarker in cystinosis. Then we'll hear from Dr. Minnie Sarwal on some work she's doing in male reproduction in cystinosis. And then we will go into our panels later in the day. So thank you so much to our industry partners, to our CRN funded investigators that were able to join us this morning. And we look forward to seeing everyone at 1 o'clock Eastern. Thank you. Welcome back, everyone. We're happy to continue on with our research update session for the morning and afternoon, depending on where you are joining us in the world. Our next speaker will be Dr. Elena Levchenko. She is the Division Chief of Pediatric Nephrology at the University Hospitals in Leuven, Belgium. She'll be talking to us today about her validation study of a new biomarker for cystinosis and a retrospective cohort from the NIH. We're happy to fund a portion of Dr. Levchenko's great work. And with that, I will hand it over to Dr. Levchenko. Elena.
Thank you very much, Christy. Good afternoon or good evening, everybody, depending on their place where you are. It's evening for me here in Belgium. And let me first thank CRN for organizing this meeting, for inviting me, and of course for supporting our project on validation of a new biomarker of cystinosis in a retrospective cohort of NIH. So I don't have to talk about this audience that cystine crystals accumulation is the main biochemical feature of cystinosis. We know that starting from the seminal publication of Jerry Schneider in '67, and we really try to decrease cystine accumulation, and all of you know that the drug we are using for that is cysteamine, which breaks cysteine inside of the lysosomes into cysteine and cysteamine mixed disulfide, which can leave the lysosome using different transporter systems, therefore bypassing their mutated cystinosine. You all know how important is cysteamine for cystinosis patient, as it delays their progression of kidney disease, and it also delays or prevents the development of extrarenal complications. And there are a lot of studies of this. I just want to remind you this very beautiful study of Dr. Nesterova and Dr. Gol, who showed that each year of good compliance of cysteamine therapy delays the end-stage kidney disease development by 1 year. And this was done based on the cumulative compliance score based on white blood cell cystine levels. In terms of extrarenal complications, again, the study of NIH by Dr. Gol and colleagues showing that each year of cysteamine therapy increased the risk of extrarenal complications such as diabetes, myopathy, lung dysfunction, many others, and actually increase the chance of death, while each year on cysteamine really delayed or even prevented the extrarenal complications. So cysteamine remains a very important drug for cystinosis patients, and actually nowadays the only drug which really interferes with the disease pathogenesis. So when we treat our patients with cystinosis, we monitor cysteamine treatment. And now we do it by measuring cysteine in white blood cells. So we isolate white blood cells from peripheral blood. And usually nowadays in the world, there are two techniques to do that. It's high-performance liquid chromatography, which you can see here, the machine for that, or tandem MS. So using white blood cells has a lot of advantages. We can easily draw blood, and we have also very good validated reference values for well-treated patients and also patients who are not so well-treated. However, we have some disadvantages. First of all, measuring cysteine white blood cells is quite laborious, and it requires expensive equipment and also trained staff, and therefore this technique is only available in a few reference laboratories around the globe, and it's not available in economically less developed countries, and for sure in third world countries. White blood cells are very sensitive to storage and transport conditions, and therefore when cells are sent from one laboratory to another and cells are not well preserved, it really can influence the value of this determination. Another disadvantage with white blood cells is actually white blood cells are short living cells. They have a life of the granulocyte, it's a cell which accumulates cystine in our blood, is only 12 hours. So when the patient takes a drug a few weeks or a few days before the consultation very well, the value can be lower, while it doesn't really reflect the long-term compliance. And another possible disadvantage that we really don't know to which extent white blood cell cystine levels reflect tissue cystine accumulation. So there is a need for probably a better biomarker of cysteamine therapy for cystinosis. So in general terms, what is an ideal biomarker? And most people say that the ideal biomarker should be sensitive when it goes about the disease diagnosis and should be specific, especially when we measure the response to therapy. It should be easy to perform and cost-effective. It should reflect abnormal physiology of the patient. It should be useful to assess the severity of the disease and the prognosis. And therefore, it should be useful in the follow-up of the patients and adjusting the therapy. So when you look with this respect to monitoring cystinosis, you think again about the main feature of cystinosis, which is the accumulation of cystine crystals, and you see these crystals in different tissues of cystinosis patients. So you can see that the levels of cystine accumulation are actually much higher in the tissues than in white blood cells when you compare it with a normal subject. What is also important for our project, that in the tissues, cystine crystals are most found in the macrophages. And here you can see the macrophage from the Bowman arrow, but actually you see this macrophage is filled with cystine crystals in different tissues of cystinosis patients. So what are these cells, the macrophages? Macrophages are cells which actually clean tissues from foreign bodies. And you can see that macrophages do that by eating, or we call this process of phagocytosis. They eat these foreign bodies, they process them, and they would expel the soluble debris to the extracellular space so that the cell can get rid of these foreign bodies. And here you can see that these kind of macrophages are present in different tissues of the people. And as I see, they have their main function is phagocytosis. They have some tissue-specific functions. We are not going to talk about them. And for cystinosis, we know that macrophages are eating or cleaning, or they at least try to clean cystinosis tissues from cystine crystals. Obviously, cystinotic macrophages fail to do that because they have the same defect that all tissues of cystinosis patients, they cannot really process cystine. And what happens that the macrophages get activated. And that was actually their PhD student who worked in our lab, Dr. Elmonem, now he's working in Cairo in Egypt. He has shown that when you expose macrophages in vitro, it means outside of the body, to cystine crystals, they can indeed eat them, they can phagocyte cystine crystals, and when you do that, macrophages get activated. So we measure this by different means, and this is one of the substances which is secreted by activated macrophages. It's a cytokine, and you can see that macrophages do it in the concentration-dependent manner. So the more cystine crystals they eat, the higher would be the level of this cytokine. What is also important for our project? I'm sorry, I have a small technical issue for turning my slides. So again, upon the phagocytosis of cystine crystals, macrophages start to secrete this enzyme, which has a terrible name, chitotriosidase. And they also do it in the concentration-dependent manner. The more cystine crystal they eat, the higher would be the level of this enzyme. And these are in vitro experiments, they mean in their outside of the body. So what is this enzyme, chitotriosidase? Just trying to move my slides and going a little bit slower. I'm very sorry for that. Okay, so this enzyme chitotriosidase is actually an enzyme which is produced by activated macrophages. Under normal conditions, without cysteine crystals, it breaks chitin, and chitin is the main component of the cell walls of fungi or some protozoa like malaria, and it protects us against these microorganisms. It is known that it is elevated in plasma of patients with Gaucher disease, and Gaucher disease is another lysosomal storage disorder. And so our hypothesis was that chitotriosidase might be useful, just as in Gaucher disease, to monitor cystinosis. So what can be the advantages of chitotriosidase? The first thing, that it is very easy to measure. You can measure it in plasma, and you can measure it in dry blood spots, meaning that you just can drop blood on paper, on the filter paper, you can put it in the envelope, and you can send it to another laboratory. So the method is cheap, easy, and you really don't need an expensive equipment, which is very important for, especially for the third world countries. And the enzyme is extremely stable, so it's very easy to store blood and to send samples. One limitation of this enzyme is about 6% of the population have a mutation, have a duplication in the enzyme, which encodes, in the gene which encodes this enzyme, and these people just don't produce chitotriosidase, meaning that in about 6% of the population we would not be able to use this biomarker. So Dr. Almonen performed first cross-sectional study in cystinosis patients, and you can see that in patients with cystinosis, the levels of chitotriosidase were remarkably higher than in the healthy subjects. In patients with kidney disease due to the other reasons than cystinosis, and it was also elevated in new cases of cystinosis. When we compare the Belgian patients, which are indicated here with black dots, and Egyptian patients, which are indicated in these white squares, you can see that there are two different populations. So in Belgian patients, the levels were lower than in the Egyptian patients. And what is different between these two populations? That Belgian patients are mostly better treated with cysteamine than the Egyptian patients, because cysteamine is not widely available in Egypt. So then after this study, we decided to perform a longitudinal study, and there was another PhD student in our lab, Dr. Wes. He recruited 57 cystinosis patients in 3 centers in Belgium, Netherlands, and Germany. You can see that among these patients, the half was transplanted and the half had renal Fanconi syndromes in native kidneys, and about the half of these patients had extrarenal complications. So the mean follow-up time of the patients was about 2 years. And you can see that these patients were rather well treated, as the average white blood cell cysteine levels were about 1.44. So using this cohort, Dr. Weiss would show that chitotriosidase allows distinguishing well and poor treated patients. You can see here that patients who had white blood cell cysteine levels below 2 had remarkably lower chitotriosidase compared with patients whose white blood cell cysteine levels was above the 2 level. So we did also some statistics, and we could show that when the chitotriosidase level was below 150, we could predict with 83% probability that these patients would have good white blood cysteine levels. So that was rather good specificity. Then we looked at the extrarenal complications. And we screened for the main complications such as hypothyroidism or altered thyroid function, retinopathy, diabetes, myopathy, and swallowing dysfunction. And we found that while white blood cell cystine levels were not associated with occurrence of these complications in our cohort, and it was a short-term cohort, the catechosidase could distinguish between patients who had one or more than one extrarenal complications. Here we had a different cutoff value of 250, and again, the predictive value of catechosidase was above 80%. So we were very enthusiastic about the validity of catechosidase to predict extrarenal complications. However, there were obvious limitations in our study. First, the number of patients of the study was rather limited. Second, we had a limited number of patients with native kidneys, and so we could not determine the predictive value of catechosidase for the kidney function prognosis. And obviously, we had a short observation period. So we really found that we needed another cohort to validate our findings before we can recommend to use catechosidase in the routine clinical practice. And this is actually the study which is supported by the Cystinosis Research Network. And this is a study of catechosidase in the retrospective cohort of National Institute of Health. We were very fortunate that Dr. Gol agreed to provide the samples of the NIH for this study, aiming to validate catechosidase as a novel biomarker of cystinosis. So, as you know, the NIH followed for years a large cohort of cystinosis patients. There are more than 150 patients followed, and there are 50 patients with a lot of repeated samples. So, using these samples, which are stored at NIH and which will be transported to Belgium, we will analyze the clinical information, the biochemical information, kidney function, age, dialysis and transplantation, of course, white blood cell cystine levels, and the presence of extrarenal complications, together with the levels of catechosidase, which will be measured in our laboratory in the samples. So, by doing that, we are going to answer the following research questions. So we are going to answer whether catechosidase really can predict kidney function prognosis in cystinosis patients. We would like to answer whether catechosidase can predict the occurrence of extrarenal complications of long term. And finally, we would like to answer whether catechosidase can be used as a long-term biomarker of cystinosis on the long-term basis. So this is a study design. We just are busy now with the paperwork. It's not so easy to arrange the transportation of samples from the United States to Europe. But it's going very well, and I hope that during the next conferences we will be able to share with you the results of this study. So I would like to end here and to thank first of all people who are working with me in the lab, especially Kunrat and Mohammed, who did these initial studies of catechosidase. I would like to thank Dr. Gol from NIH and also Galina Nesterova for agreeing to perform the study and to share the clinical data of the patients. I would like to thank their Cystinosis Network Europe, who are always very supportive, and also Cystinosis Ireland, who are very supportive to all studies we are doing. And of course, I would like to thank the Cystinosis Research Network for organizing this meeting and for supporting our project. So this is the flute map of Belgium. You can see that the red regions are the regions which are really affected by terrible floods. We were very fortunate in Leuven. You see Leuven here, and you can see that the water came very close to our door, but we are just very lucky that we did not have water in our houses and also in the hospitals. But it happens only once in a lifetime in Belgium, so normally Belgium is very safe. country to visit. And I would really like to invite you to the cystinosis family meeting, which we are planning to organize in Belgium next year. So here are the dates of the meeting. And I really hope that many of you would be able to join us in Belgium. And there are a lot of good things here, like good beer, good chocolate. And I hope to meet you all in Belgium next year. So thank you very much. And I'm happy to answer your questions.
Thank you, Elena. That was a really fantastic overview. We're really happy to support your work in conjunction with so many other people. We do have a few questions that have come in, and I'm grateful that you're able to join us here live to answer some of these questions. I think I should first say that I think your work with Dr. Gall and Dr. Nestorova and the samples you're able to leverage from the NIH is a very good example of how our community can be grateful that their participation is being utilized not only beyond the treatment that you get at the NIH, but there's collaboration that takes place within all of the folks that are interested in cystinosis in the world. So thank you for that. And then further, the biomarker study, we all know that white blood cell cysteine level testing can be difficult at times. So I think this work is going to be really important. We've had one question come in, and I'll read it to you and let you answer. It says, with this modality of obtaining this sample, would it be given or obtained via venipuncture or through a blood draw, or could it be as simple as a finger stick? And could it possibly be done in a home setting versus the hospital? And I'll let you answer.
Yeah, that's a very, very good question. Actually, the answer is that I don't know that. So Dr. Enwola here compared values in plasma and in blood spots, which were generating by dropping their venous blood into blood spots on the filter paper. So it is possible that the value which we can derive from the capillary blood, and this is blood that we get when you just take blood from the finger, it can be slightly different. So I think that would need validation, but I would think that it would be possible, but I think we will need to measure that. Of course, it would be beautiful if we would be able to measure just as people measure glucose in case of diabetes, but I don't know yet the answer for that.
Thank you. That's actually a great answer. But I think the advantage to that would be the easier that we can make it for patients to have their white blood cell cysteine levels done. And then this new biomarker, even better, that we'll be able to judge how treatment is going so much more easily. So I have another question, and this is sort of a practical question, and I have a feeling you won't be able to answer this, but folks are wondering what the timeline would be for something that's, you're looking at so early to the point where this would be available in clinics to patients.
Well, I guess that actually in Belgium, we are already using Kaito because we were quite enthusiastic. So we are actually, in our hospital, we are using it already in the clinical practice. I must say that I would not really rely too much on that before the findings are validated. But we have now a study going on also in Belgium to see whether we can already go a little bit closer to the clinics. But I think to demonstrate the validity, we will await the result of the study. Of course, the study is a research, and so we will see what the answer would be. And then I guess that after that study, the study would last actually 1-2 years before we have everything done, calculated. Then in this time frame, we would be able to conclude.
Okay, thank you for that, Elena. So that is the end of the questions that I have submitted. We're so grateful that you were able to join us live today and address some of these questions. We thank you for your work. And we hope that everyone who's able from around the world will join us in Belgium in 2022 and be live with our community. So thank you, Dr. Levchenko, for today.
Thank you very much, Kristen. Bye-bye, everybody. Bye.
Bye-bye. Okay, so we're going to move on now to our final speaker for today. That is Dr. Minnie Sarwal. She'll be talking to us about perturbations in the VATPase pathway drive pathology in the male reproductive system and cystinosis. And this work that Dr. Sarwal and her lab is doing is not only supported by CRN, but is supported in a joint funding way with our partnership with Cystinosis Ireland. So I just wanted to note that our friends in Ireland are also supporting this work. Dr. Sarwal was previously medical director of pediatric kidney transplantation at Stanford University. She is currently the director of precision medicine translational program and solid organ transplantation at the University of California, San Francisco. We're so thankful to Dr. Sarwal for submitting her presentation for us to view today. And so with that, I will present to you Dr. Sarwal's work.
Thank you for this invitation to the Cystinosis conference. I'm delighted to be presenting here virtually some of the work that we've been doing in our lab at UCSF using transcriptional profiling methods to understand perturbations at the gene expression and protein level that drive the changes in nephropathic cystinosis in the kidney, and extending some of these understandings to what may be overlapping pathways that may also be driving problems with the male reproductive system. And I'd like to talk about a new finding about perturbations in the vacuolar ATPase pathway that we believe may be responsible for some of the pathology in the kidney as well as the male reproductive system in cystinosis. Here are my disclosures. And I will not be talking about anything which is in conflict or proprietary in this presentation. There are a few unanswered questions in cystinosis, which relate to the following problems that occur with regards to injury to the kidney, known to occur as the Fanconi syndrome, and then renal failure requiring transplantation, as well as problems with male infertility, hypothyroidism, and muscle weakness, just to name a few, because of course cystinosis is one of lysosomal storage problem, which of course is present in all tissues and organs systemically. But there are two major unanswered questions that affect us as nephrologists. Number one, cysteine depletion does not reverse the renal Fanconi syndrome. And even in children where cysteine depletion therapy has been started at birth, we have been not been able to avoid observance of the renal Fanconi syndrome. And secondly, cysteine depletion, when taken regularly and especially started very early in life, can delay the onset of renal failure and move it from the first to the second decade of life. But this does not prevent eventually the onset of renal failure. And why not? We had done some earlier studies which have been published where we looked at microarray analysis of peripheral blood mononuclear cells from patients with and without cystinosis, and extended some of these studies also to looking at key pathways in the kidney biopsy tissue from patients with cystinosis. And we uncovered a host of pathways that relate really to established cellular injury that's occurring in these patients with regards to key pathways involved in cell death, cell cycling, amino acid metabolism, and autophagy, as well as changes in the expression of this marker clustrin with both intracellular clustrin and soluble clustrin with the kind of ratio changing in the course of cystinosis. But we now believe that these kind of changes that we see are just a response of the tissue injury and the cellular injury to the process of the lysosomes being sick, and is really a readout of that kind of injury, but didn't lead us to actually understand truly the pathogenesis of what may drive this. So to understand this further, we actually did global transcriptional analysis using Affymetrix microarrays, using renal proximal tubular cells that were isolated from urine samples of patients with and without cystinosis, as well as from cell lines that were actually loaded with cDME to try and see if this would mimic the cystinosis disease, as well as from fibroblasts from patients with cystinosis to try and understand if there was actually commonality in the injury that occurs in the cell in cystinosis, irrespective of tissue source. So these studies were first started by Poonam Sansanwal, a postdoc in my lab, and now continued by Swastika Sur, very talented postdoc, who will also be on the call at the end to answer any questions that arise. So first of all, we could understand in this principal component analysis, if you look at these orange samples, which are each of them is a unique patient with cystinosis, we can see that the distance of these samples from other samples suggests that they're actually quite different at a gene level or transcriptional level. And you can see that these dark orange samples are actually normal renal proximal tubular cells. So the patients with cystinosis have very different signatures from normal people. But importantly, when you take those normal proximal tubular cells and you load them with cDME, you can see that their profile still looks very similar to normal, not really like cystinosis. Also importantly, when you actually take fibroblasts from the same patients with cystinosis and you profile them, you see that they don't really look a lot like the renal proximal tubular cells from the cystinosis patients, but in fact, they look closer like the normal fibroblasts. So the tissue looks closer to itself, and actually cystinosis looks more disparate. So cysteine loading, we believe here, shown in green, is actually a very poor in vitro model surrogate for cystinotic renal injury, and it's been used previously to try and study the disease, but we felt that probably need to pay attention to finding other in vitro models, and Swastika did just that, and I'll show you that in the following slides. And we also see that the gene expression changes in cystinotic disease are really very specific to the kidney. And you can see these changes that we see here are shown at various pathways relating to DNA replication, oxidative phosphorylation, cell cycle analysis, etc. So if we actually look at what was one of the most important pathways that came out of this analysis, was a multi-gene family of genes belonging to the vacuolar ATPase pathway. And you can see that all of these genes were actually downregulated in cystinotic renal proximal tubular cells. You can see this VATPases are actually a large subunit complex. It functions for driving ATP-driven proton pumps, and basically helps to acidify intracellular compartments and transfer these protons across the membrane. It consists of a V1 domain, which is involved in ATP hydrolysis, and a V0 domain involved in proton translocation. Very importantly, this gene family is very important for renal acidification, bone resorption, homeostasis of cytoplasmic pH, as well as sperm maturation. Different members of the family are potentially involved in these different functions. But the fact that a single gene family was involved in both addressing renal acidification and sperm maturation caught our attention to try and create this link in patients with nephropathic cystinosis. The other important thing is that different VATPase isoforms are present, which may actually be perturbed in disease. And this is something that we are exploring. But they are supposed to be tissue specific. But importantly, again, the link between the kidney and the testes, you can see that these different isoforms are actually commonly expressed in both the kidney and the epididymis, which is a region of the male reproductive system. So if you look at these vacuolar ATPases, our transcriptional profiling identified that they were all significantly downregulated in the cystinotic kidney. But we showed that in addition to the gene level, they were also downregulated at the protein level. And actually, when you did immunohistochemistry of renal proximal tubular cells, you can see a marked difference with a much lower amount of green, really, staining here, showing much lower amounts of VATPase also at the protein level. Now, since the VATPase is involved in acidification, our hypothesis was that therefore there would be less acidic intracellular pH in cystinosis. And here Swastika shows quite nicely that actually when she did these studies, there is a significant difference in pH between cystinotic and non-cystinotic. And then if this is relating to some of the injury pathways that we had seen in the peripheral blood mononuclear cells, the first set of experiments that we did, and also reading the literature, there is a change in mitochondrial function, energetics, etc., increase of reactive oxygen species because of injury to the cell. So do we see mitochondrial ATP production being perturbed in these patients? And of course, you do see that, which you would expect. And you can see in cystinotic RP-TEX, if you actually add the inhibitors to actually show if the OCR amount increases, you can see it doesn't increase to the same level, and ATP production is lower. So we already addressed that CDME loading was actually a poor model to study in vitro cystinosis. So Swastika set about actually creating a new in vitro model by taking the CTNS gene, knocking it out by CRISPR-Cas9 in the cell line and of RPTEX, and replicating to see whether this model that she had created could actually replicate the same changes of the VATPase genes, as well as the same phenotype as we were seeing in the primary renal proximal tubular cells extracted from patients with the disease. So the first is, if you actually created the CTNS knockout cell line as a model to study, it does result in cysteine accumulation. It has a lower amount of VATPase. The VATPase is reduced at the protein level. It shows the same changes in pH. It does show the same changes with the mitochondrial ATP production. So it was a very important surrogate model that actually could be used to study cystinosis disease in vitro. And if we actually look further, we can see that overexpression of VATPase is actually also causing mitochondrial dysfunction, as well as increased autophagosome turnover. So you can see this in the study, looking at a ratio of LC32 to LC31. I can see in control versus in the VATPase, if plasmid is given, you can see that there is actually reduction of the autophagy. And so to do this is CTNS knockout cell line was taken. This is the control, and the VATPase plasmid was actually transfected. And you can see that restoring VATPase levels actually reduces the autophagic injury. And also it results in some rescue of the mitochondrial ATP production. And importantly, if you actually then look at the cell and you look at it by EM, you can see that in the knockout, there is lower amounts of mitochondria. If you add back VATPase, you're seeing the rescue. And similarly here, the mitochondria in the knockout look actually quite unhealthy. And you can see if you restore the VATPase here, you actually get improvement in mitochondrial cristae as well as function here shown above. So this was an important molecule that seems to be related quite directly with the injury that we are seeing here, independent of just cysteine loading. So to try and address if we could do something with increasing VATPase levels in cells and see if this could rescue the phenotype, we basically looked at an FDA drug database that's been developed by our lab and looked at key pathways that were dysregulated in cystinosis and looked for drugs that actually affected these pathways, including VATPases. And we found a compound, which we call MMS 1269, which actually was found to increase VATPase expression. And this is looking again now at the CTNS knockout RPTEX. And so you can see with the drug, you can see the VATPase amount is increasing, and you can see the LC3 expression is decreasing, showing improvement of autophagosome turnover. It's also improving mitochondrial membrane potential. Here is the human immortalized control. Given the compound, you can see no real change. Here's the CTNS knockout, now given the compound, and showing really very nice rescue of mitochondrial membrane potential. And again, also showing a reduction of when the control cells as well as the knockout cells, which actually have a lot of mitochondrial ROS. When we're treated with the compound, you can see there is a reduction of mitochondrial ROS. And here the data has been normalized to live and dead cells. So a schematic overview of normal and cystinotic RPTEX therefore shows that we've been able to learn these areas in blue. We've been able to understand how essentially you have the problem with the CTNS downregulation, absent or dysfunctional protein, which basically somehow links and results in this lower VATPases with loss of normal lysosomal acidity and dysfunctional mitochondria. We've also got some other genes that are being affected and downregulated, which involve neutral and heterodimeric amino acid synthesis, which also results in this kind of lysosomal injury. We believe that the earlier pathways that we looked at for increased inducible clustering levels, you can see is also probably a net effect that is leading to increased autophagosomes and dysfunctional mitochondria fusing with the lysosomes, which is kind of shown here. So this little cartoon puts together some of the new learning that we're getting of how some of these injury pathways are occurring. But the VATPase in itself seems to be driving a lot of these functions that are relating to the lysosome, leading to an autophagic flux block and leading to dysfunction directly of the mitochondria. So as we looked at these microarray studies, and we started to understand more about cystinotic RP-TEG injury, we saw that some of these genes were actually also highly expressed in the testes, and that made us think again about how there could be overlapping pathways that are present here and genes that are defective and driving both the kidney injury, but also the injury to the testes. So to propose studying this further, we have put together a funding application where we will be looking at perturbations in the ATPase pathway and understanding how these drive pathology in the male reproductive system. This has been jointly funded by the Health Research Board of Ireland, Cystinosis Ireland, and the Cystinosis Research Network, and it'll be conducted by Swastika Sur in my lab, Polina Lishko at Berkeley, as well as James Smith at UCSF. And we are proposing basically to first do a clinical enrollment of patients and then collect biosamples at semen and gonadal biopsies. These will be all done by Dr. Smith, and these biopsies of the epididymis and the testes, as well as sperm samples. We also, in a collaboration in the study with Dr. Levchenko at Leuven, expect to receive a few testicular biopsies also from patients with nephropathic cystinosis. The tissue will actually be processed for single nuclear RNA-seq as well as for codex for spatial multiplex analysis to see if you're identifying new cell types and cell states in people with nephropathic cystinosis and male infertility. And then also we'll be looking at the role of cTNase and VATPase in the sperm acrosome, because in case this is resulting in problems with non-obstructive azoospermia and decreased motility. We also, to try and support this work, applied for a SeedCon grant, which again, Swastika got funded, and she has, in fact, is halfway through creating the cTNS knockout now but now in Sertoli cells isolated from testes, again, using CRISPR-Cas9. These cells are being immortalized, and then a cTNS guide RNA is used for knockout of exons 3 to 12. Here, Sanger sequencing is showing that we're getting some about total efficiency of about 86% for knockout purity. Repeat Sanger sequencing will be done after single cell purity, and then cysteine accumulation will be tested in the knockout by HPLC. You can see we have some preliminary data coming from testicular biopsies. Here we only have one patient with cystinosis so far, a sample that was supplied to us by Dr. Levchenko, whereas we have 6 samples of patients with male infertility provided to us by Dr. James Smith for causes other than cystinosis, and comparison of gender reconstruction and vasectomy that happen where small samples are also available from fertile males. So it's important to see, first of all, cTNS here, as you can see, is actually absent in patients with cystinosis, so which we expect. CFTR, which we know low to be low in cystinosis, is basically lower in these patients. And then you can see that there are a bunch of VATPases that are also lower in cystinosis. These are actually VATPases that are not involved in the kidney and only seem to be involved in the testes. And in here you can see other kidney-specific VATPases that we spoke about earlier. Actually, they do not show that decrease. In fact, seem to be increased in the testes. So there is a lot of work that's going to be done here to understand the role of these VATPases being severely downregulated in the testes and how this kind of correlate with the pathological occurrence of male infertility. So we have an invitation to participate. This clinical characterization and biosampling that will be conducted. The first part of it will be an observational study to understand the structural, hormonal, and molecular alterations that result in the development of infertility in cystinotic males. Dr. Smith is director of the Male Reproductive Health System at UCSF, and he has done a countless number of these procedures and purports a lot of safety as well as low morbidity in doing any of these procedures and biosamples. We have funds in place to support travel of people to come to UCSF to actually have the observational study and the biosampling. And once the pandemic opens up, Dr. Smith and I plan to travel to Europe to actually go to a few select sites where we will be able to actually also meet with patients and potentially complete the questionnaire and the biosamples. So if there is interest to participate, please do email me at mini.sarwal@ucsf.edu. I'd like to acknowledge really now all the people in my lab. There is Swastika here, as well as all the rest who have done an enormous amount of work to support the work that is presented today, and our collaborators as well as our funding agencies. Thank you very much for your attention. I look forward to taking any questions. Thank you.
Thank you, Dr. Sarwal, for that great update on the really important work that you're doing on male reproductive issues and cystinosis. Any questions that anyone might have for Dr. Sarwal, please submit them either directly to Dr. Sarwal's email, as she noted. You can also, as with all of our presenters, submit questions to CRN, to me directly, or to the info@cystinosis.org email address. We thank Dr. Sarwal for her work. We thank Cystinosis Ireland for their co-funding of that work. They're great partners for us to work with. So with that, I'd like to conclude our morning and afternoon sessions with updates from our industry partners and our funded researchers. It's really amazing to hear about all the great work that's going on. We will hope to make some of the recordings of the sessions from the entire conference available via our YouTube channel. Stay tuned for more information on that in the coming days. And then several of our speakers have talked about research opportunities. As always, we'll be passing those opportunities along to the community via the website, our newsletter, and our social media channels. So we'll be sure to keep everyone updated. Next up are our final two panels for the day. Those will look more like the traditional Zoom meetings that you're used to. You'll be able to see the other participants. You'll be able to submit questions via the chat and actually unmute yourself and interact. So those panels will begin at 2 p.m. Eastern time, so at the top of the hour in about 13 minutes. You'll need to log out of this session and then go into the attendee hub and log into those panels. First one will be the parents of children and adults living with cystinosis panels. We have a great group of folks that are willing to share their experience as a caretaker and as a parent, both at the beginning of their journey with cystinosis and then farther along. We have a parent of an adult with cystinosis that will be joining. And then finally, at 2:45 Eastern, there'll be a panel session that will take place that will involve some of our members of our A-Lab group, so adults living with cystinosis, and those adults are active on our A-Lab group, and they have been generous enough to meet us and share some of their personal experiences, and we'll be there and available to answer questions. So with that, I'd like to thank everyone for joining us today. We had lots of people from all over the world, and we will see everyone in about 10 minutes via the attendee hub at the first panel. Thank you so much.